For 20 years, they have done research, and now they found the most successful supplement to increase longevity. They mean the ITP, the Intervention Testing Program, the most prestigious longevity research organization today. Which supplement did they test? Astaxanthin. Astaxanthin is a molecule made by algae and actually gives salmon and flamingos their pink color. So if you want to live longer and stay younger, should you just go and buy Astaxanthin as a supplement form and take it? Not so quickly. If you've been following my channel, you know that the devil is in the details. The dose, the timing, cycling, and the brand. Which brand did they use in the ITP study? And can you buy it today? The answer is yes. What I've done for you today is tracking and finding the exact manufacturer that provided the ITP with their product. This person, Dave, has been researching astaxanthin for over 25 years, and he's probably, in my opinion, one of the top 3 experts on astaxanthin for longevity in the world today. Especially in the context of longevity, because he shares this passion as well, just like me and probably like you. So it's part of our community, to be honest, and he's much more a researcher than a salesperson, as opposed to other people who sell supplement and actually are very biased about what they sell. So Dave Company supply the ITP with the material, and he will tell you everything you need to know about the research on astaxanthin, the side effects. We are going to discuss the impact on your exercise routine or your testosterone, how much astaxanthin he takes personally, and what's the best way to translate in the dose from what they achieve in the ITP study to our bodies. This has shocked me because I would never guess this translation. I actually made a mistake and he corrected me. Before we started this podcast interview, there are 2 disclaimers. The first one is I'm not affiliated with any supplement company. I don't care what you take. I want you to stay healthy, I want you to stay young, but I don't really profit nor do I care about which supplement you buy. The second disclaimer is this is the most in-depth interview on astaxanthin today on the internet. It's long, but I promise you this, it's extremely practical, and if you want to live longer, that you need to know all the small details in order to form a habit that makes sense, that is actually effective. So I suggest, despite the length of this video, give it a chance and listen to everything. And if there is a segment that is not interesting to you, use the chapter to go to what is interesting. Now enjoy. Astaxanthin, the most exciting supplement for longevity on the market today. So today we're going to speak with Dave. He's the founder and CEO of AX3 Life, which is the astaxanthin that has been used by the ITP in 2024 study that has been shown to increase lifespan in male mice by 12%. So I've read 2 lifespan studies that increases lifespan in worms, and 2 lifespan studies it increases lifespan in fruit flies, even though under some stress, some oxidative stress. And also we have the FOXO activation, which we know works on humans as well. So this was like the background to the ITP. Pre-ITP, there was any piece of evidence that you feel is super important besides the one I mentioned that is relevant for longevity specifically?
There was yeast as well, which just contributes. It's not specifically, a major addition, but that's also part of the preliminary research that was published, I think, prior to the ITP starting, or at least during the ITP, certainly prior to the ITP study being completed. But I think apart from those key studies, there's the mechanism of action types of studies where you look at astaxanthin's impact on important pathways related to aging. So AMPK and impacts on mTOR, the sirtuins, FOXO3, NRF2, et cetera, et cetera. So I think all of that helps to contribute to the understanding that it makes sense that it would have an impact on longevity, other than just the empirical evidence that it did extend longevity in those model organisms. So that would be probably the best evidence that we had. There's certainly nothing in humans. It's very hard to test longevity in humans. To follow people for decades, anecdotally, that's also hard to know, looking at people's diets in longer living regions of the world, et cetera. Do they contain astaxanthin? In the wild, some of the long living sea creatures do consume a lot of astaxanthin in their diet.
Whales,
so that contributes. Yeah, exactly. Whales and crustaceans and fish and so that also helps. But I think that's the majority of the evidence that gave us the understanding that let's see if we can demonstrate this in mammals, because the ITP was the first mammalian test of astaxanthin for lifespan. And the ITP, I'm sure everyone listening has heard of the ITP to some extent, but if not, it's an NIH funded program specifically by the National Institute on Aging. And that's part of the United States National Institutes of Health, which is the largest funder of biomedical research in the world. And this program is conducted at 3 independent research organizations at the University of Michigan, the University of Texas, and Jackson Labs in Maine. And they essentially run the study in triplicate in parallel. And so they have like 3,000 mice each year that are genetically heterogeneous. And so they're more translatable to humans based as compared to the typical laboratory inbred strains of mice that are used for a lot of basic research, but would not necessarily give us the best translation to real world, human populations. So they have these, specially bred, genetically heterogeneous mice.
How long have you been doing those studies? Like 15 years, maybe?
20
Or
years.
12?
Yeah, the program
20
started.
years. And this is today, 2024, came up with a study that showed that astaxanthin is the longest extension in male mice in all 3 locations. I think this is pretty astounding. And people need to understand the importance of the study, because I think the astounding thing is they found 2 molecules that extended lifespan in the study. One of them was a drug. This drug only increased lifespan in males in 2 locations, but the astaxanthin was very consistent in all 3 locations, all 3 different labs. It increased lifespan, and I think it's pretty astonishing. And I believe it's like the longest extension they've ever had for a natural supplement we can buy off the shelf, not like a drug such as rapamycin. It's not.
Yeah, because fish oil has been tested, curcumin has been tested, resveratrol has been tested, fisetin has been tested. Other drugs like metformin, NAD precursor NR was tested. And so a lot of things that have a lot of interest and merit for health and longevity have been tested. But to your point, this is the first time in the 20 years that a naturally occurring molecule that's a supplement that's widely accessible, exceptionally safe, was shown to increase lifespan by more than 10%. In fact, like you said, 12% with very high statistical significance. I think the p-value was like 0.003. So very, very strong statistical significance as well. And so, that's a really big deal, because otherwise you had agents like rapamycin that, performed really well from a lifespan standpoint, but from a real world standpoint, not necessarily something you want to take every day. And so people are looking at different dosing regimens and analogs for rapamycin. But you want ideally to have something that can extend lifespan, extend health span, and be something that you could reasonably take every day. So this is a really big deal, like you said, after 20 years with this incredibly rigorous independent program.
Are you all aware of the specific cause of death that was reduced by astaxanthin? Because I know 80% of these mice that die from cancer.
So the mice in this model typically die from a cancer of one type or another, and that's just the thing that is the cause of death in the mice.
Like that,
whatever it may be. There's different types, but essentially the aging is manifested in the form of cancer. Like they're not getting heart disease, but they're getting cancer, and that ultimately is the reason for cause of death. But it would be, one could say, oh well, therefore this is a cancer treatment, but it's more, the theory is that it's treating the mechanisms of aging that ultimately are driving, in this case, aging or cancer in the mice, whereas in humans that may be different. With that said, there are mechanistic rationales that astaxanthin impacts heart health, and there's research related to cancer. Again, you can't make claims like that as a supplement, but there is cancer research being done with astaxanthin, some interesting results there. So I don't think, it's not like it was a cancer agent that was being tested, but it was something that mitigated or slowed down the aging process that ultimately manifested itself as cancer in these mice.
It delayed the cancer in the male group, right? Is this correct? It delayed the cancer.
Yeah, I mean, they didn't measure cancer specifically, but certainly it delayed their death, it prolonged their lifespan, and so presumably that was based on cancer for the most part. Across
and they did
all
not
the
measure
mice,
health span. They did not measure health span, right? That's what they're looking for.
They can't. It's just not. It's not practical for them to do that.
I wanted to know, with the female, if I could.
And the females, I'll mention the females had a 3% increase in lifespan, but it was not statistically significant. And so, but the females already live about 9% longer, and so ultimately, when you look at the total, duration of lifespan in the males and females, they were just about the same. The females were slightly longer, longer lifespan, and so I think that it essentially allowed astaxanthin to have the males catch up to the females in terms of lifespan. But why there was the difference, we don't know specifically. Something you would presume related to hormones, et cetera. And so I think that would be interesting research. But it would be incorrect to say that, well, therefore this is only a benefit for men or for males. This is
without
something
health
that
span, without health span. But I also,
we have health.
When I look at the graph, I saw it was extended in the females as well.
Visually you can see that it extended a bit,
Yeah.
and numerically it was, although not statistically significantly. Maybe if you had a bigger group, potentially. But again, the females already lived longer, and so now with the astaxanthin, the males lived just about the same as the females, both being slightly longer than control. And so it might be that just there's a maximal extension effect, and the females already were there, or maybe there's something else, regarding females and the aging process that astaxanthin has better impact for males. But from a health span, while health span wasn't tested, from an overall health benefit, like the study we did in cardiovascular subjects, that was males and females. And other clinical studies are done in males and females, animal research,
the
males
FOXO
and
activation,
females.
The FOXO activation worked both male and female?
The mouse study with UH, I don't recall if that was males or males and females. So we'd have to double check on that. But certainly the totality of evidence shows that there's benefits in males and females for various applications of health, cardiovascular, cognitive, et cetera. So I think it certainly can help males and females in terms of overall health in specific areas of health. And then in terms of actual lifespan, certainly males, and then we'd have to see with females.
In the next section, we will cover the mechanisms of action of astaxanthin, which is very important because we need to understand how can astaxanthin mechanistically increase lifespan and longevity. There are two mechanisms Dave and I are going to discuss. The first one is unique antioxidant effect, which correlates with freezing time. You're going to discover that in a second.
Let me go to the chemical activity of this compound because it's very interesting to me. In my perspective, when you want to preserve your youthfulness, you want to freeze time. You don't want to change, and you don't want your membranes to change all the time. And my understanding, correct me on this, but astaxanthin is a bit unique antioxidants in two aspects. One, it can have multiple hits of free radicals. It won't just gonna get one hit from the free radical, then it's gonna die. It's gonna have multiple cycles. And the second is it cannot become pro-oxidant. We know if you, for example, put too much, even outside, if you put too much vitamin E into fish oil, it can oxidize the fish oil. It makes me more wary about antioxidant recommendations, but it would seem that astaxanthin doesn't suffer from these two things. So it's more like a long-lasting antioxidant that stabilizes your membranes, and it fits more into the picture that I have of freezing time, which I want to do. So can you correct me on these two aspects of the chemical properties of this molecule?
Yeah, no, you're definitely correct in terms of astaxanthin being a very unique antioxidant. And so a lot of people think of all antioxidants as being the same, but they're not. And so the structure makes a really important difference. And even within the carotenoid family, if you look at astaxanthin versus beta-carotene or lutein and zeaxanthin, they look to the untrained eye very chemically similar from the molecular structure. But like I mentioned earlier, astaxanthin has oxygenated head groups with hydroxyl OH groups and also ketone groups on each ring moiety at the end of the molecule, whereas beta-carotene does not. And so that allows it, like you said, to anchor across the membrane and stabilize the cellular membrane. And we've done work with a collaborator of ours who has an appointment at Harvard, but also his own private research company where they had a biological membrane model, and they incorporated astaxanthin or other antioxidants in and looked at the electron density across the membrane. So you have a higher concentration of electrons at the phospholipid head groups, and then a lower concentration at the tails. And you can look at that electron density across the membrane. And when you put astaxanthin in, that density or distribution of the electrons was virtually unchanged. And if you incorporated other things like beta-carotene or even lycopene, you saw a very significant disturbance in the electron density and disturbance of the membrane. And so it is stabilizing the membrane.
What does it mean, disturbance, so people could understand?
So the membrane is comprised of a lipid bilayer. So you have phospholipids, kind of two back-to-back lipids with the heads and the tails, kind of in opposite directions, and that comprises the membrane. When it gets disturbed, those are no longer nicely ordered. They can get, kind of angled in different directions, and you don't have a nicely ordered, stacked membrane of your lipids nicely, laid out across the membrane, and that can have a lot of negative effects on cellular function if your membrane is depolarized or disturbed. And so astaxanthin helps to maintain the integrity of the membrane as compared to other antioxidants and even other carotenoids. But also with that, it has the ability to scavenge free radicals both inside and outside the membrane, because it can trap free radicals with its head groups, but also which would be at the surface of the membrane inside and outside, and also inside the interior of the membrane with the double conjugated polyene backbone of the molecule that can also quench free radicals. And so it has the ability to fight free radicals in and outside of the membrane. And then it can say regenerate by interaction with vitamin C, which can regenerate vitamin E, for example, as an antioxidant. So it can play well with the other antioxidants as well. And so I think with astaxanthin, you have that unique capability based on its structure to not become pro-oxidant, which, by the way, in that model membrane system, not only did it stabilize or maintain the integrity of the membrane, lipid peroxidation was measured, and lipid peroxidation or oxidation of the lipids in the membrane was reduced with astaxanthin, whereas it was increased with beta carotene, which was acting as a pro-oxidant in that case. And so that's a really cool study that we collaborated on.
What about the others? Lycopene, also did they measure other?
Yes, all of the other ones had varying levels of membrane disturbance and lipid peroxidation in that particular model, whereas astaxanthin was the only one that had the reduction of the lipid peroxidation. Wow. And it's not to say that all of those are bad antioxidants.
So it's almost they attract a fire to the membrane.
I mean, potentially, yeah, that could be one way of putting it. Or they disrupt the membrane, which is not good, and it potentially can be pro-oxidant. And so astaxanthin, I think, is very unique in that case. And so, but in addition to that, there's been some studies in test tubes looking at like singlet oxygen, scavenging, quenching. That is, one measure of its kind of antioxidant capabilities, and it actually was much more effective at fighting the free radicals compared to other common antioxidants. And it was shown to be like 6,000 times stronger than vitamin C, or 550 times stronger than vitamin E, and like 40 times stronger than beta carotene. And so from a scavenging standpoint, it appears to be very effective. But I think almost more importantly is the fact that it localizes in that special way in the membrane, which is very unique. And the fact that it not only gets to the outer plasma membrane of the cell, but like I said, gets to the nuclear, mitochondrial, and other membranes in the cell, which are all important places. And if you can do that, you can reduce the oxidation of the lipids. But if you're at the nuclear level, for example, you're preventing hopefully the oxidation of the DNA and then ultimately proteins. And so all of these things have important impacts on all of your cellular signaling pathways and functions. And so there's benefits to mitochondria and other cell signaling pathways as well. And so these are all ways that astaxanthin can benefit. But at its core, it's sitting in the membranes, fighting free radicals, stabilizing the integrity of the membranes. But then I think it has all these downstream, this cascade of benefits on inflammatory pathways and other signaling pathways.
My understanding is correct. When it comes to the cell, this is my imagination. So I see a circle. It sits one side of the membrane coming in contact with the blood, the inside coming in contact with the cytoplasm, and the same idea with the membrane around the mitochondria. So the same idea. So these are like the four locations that I see in my mind that it sits. Is this correct? And also, is it dispersed also in the cytoplasm as well?
No, it could, because it's lipid soluble, and so it'll be in the membranes. It wouldn't be in the intracellular cytoplasm because that's an aqueous environment. And so, but it would be in the membranes, but not just of the plasma, the outer layer of the cell, and the mitochondrial membranes, but like the nuclear membrane as well, and the membranes of the Golgi, the ER, the other cellular organelles. And so I think it's in all these important locations. And even though it's not in the cytoplasm, it's in all these key places where important activities are happening in the cell.
And how could it absorb so many hits compared to? So in my mind, you take vitamin E, it goes into the membrane, it gets oxidized, and then the body has to get rid of it some way. When it's oxidized, may even attract some free radicals. So to me, when I take a vitamin E, I'm not really sure the behavior of the vitamin E is going to be very unpredictable. So how is that different from astaxanthin? And how come astaxanthin absorbs so many hits?
Well, astaxanthin, if you look at the structure versus vitamin E, is different. Vitamin E doesn't have that second, as long of a polyene conjugated, backbone or a second head group, and so it anchors into the membrane but kind of floats around. And so I think that astaxanthin has more areas on the molecule to fight free radicals, but also I think that potentially can be regenerated by, say, like vitamin C, where that can, like with vitamin E, could potentially help to regenerate it as an antioxidant. But
regenerate
that's also why.
So it would.
Oh,
it
yeah,
would prefer,
like with vitamin E.
It would defer. It
Yeah.
would defer to astaxanthin. Do you think the vitamin C and E
Well,
would defer to astaxanthin? Yeah.
I don't know about vitamin E interaction with astaxanthin, but say with vitamin C, like for example, with vitamin C and vitamin E, they can play together, work together. That if vitamin E becomes oxidized, then vitamin C can help to restore that. And so with astaxanthin, potentially it could act in the same way. And I don't know of research specifically to that effect, but that would be more the hypothesis, that it could transfer those electrons and potentially be regenerated. But also, if you are taking consistent daily dosing, then hopefully you are restoring astaxanthin in the membranes as needed. And so I think, again, that mechanistically it'd be really cool to see more and learn more about that. But that understanding was something that we were thinking about over 20 years ago, because when we were first doing the pharmaceutical research and we were doing those amino acid derivatives and other derivatives of astaxanthin that I was talking about that help to increase the water dispersibility and absorption in the gut, we actually were looking at an astaxanthin disuccinate, so esterified with succinic acid, but then also we had vitamin C molecules attached. So it was an astaxanthin disuccinate divitamin C conjugate, and the thought process there was that we'd be delivering astaxanthin with vitamin C to help it be performing at optimally. And so that wasn't based on actual research looking at dosing with and without vitamin C, but it was based on the mechanistic rationale that potentially vitamin C could help it. But I think just generally speaking, just the molecule itself is better constructed to fight free radicals. It has more places to do that.
Now, Dave is going to discuss another mechanism of astaxanthin, its potential activation of longevity genes.
I tell in my group that it speaks to the DNA. The DNA sees astaxanthin. I speculate because the algae, you make astaxanthin by exposing the algae to some stress. For whatever reason, I think our DNA has a read on it, and it says, okay, there is some stress, I'm going to activate these longevity genes. In my mind, the activation of longevity genes is separate and distinct from the antioxidant defense, which also beneficial because of the various properties that it has that is different. Why, from all the astaxanthin products on the market, the ITP has chosen your product, the Cardex patent astaxanthin, over the other products?
Well, it stemmed out of research that we did with our collaborators at the University of Hawaii, who are renowned geroscience researchers, and they had a model looking at FOXO3 expression in mice. And based on the preliminary research with astaxanthin that demonstrated that astaxanthin extended lifespan in the C. elegans worms and fruit flies and yeast. And so in the C. elegans, there is a particular gene that they have that is essentially their version of the FOXO3 gene that in humans is correlated or known to be associated with longevity.
I'm just translating to the audience. So the FOXO is basically one of the two genes that have the biggest evidence for extending lifespan in humans, not just animals. So this gene is really important. It's not just any longevity genes, it's like it's like very high confidence longevity gene, correct?
Exactly. And so a lot of that actually was elucidated from the Honolulu Heart Program, which was a study here in Hawaii that followed thousands of Japanese American men, from like the 1960s, onward, and looked at all of their doctor's visits, the reasons they got sick or passed away, collected blood and tissue samples, and it's this amazing database of that researchers can utilize to explore health and longevity. And one thing that emerged from that program was the role of FOXO3. That we all have the FOXO3 gene, but there's different versions of it that are
Let me ask
more
a tiny
activated.
question. Is that correct that the FOXO controls the IGF-1 insulin pathway? This is what it affected by. In essence, the insulin IGF-1, they speak to that gene. Is that correct?
I believe so. I'm not an expert in the FOXO3 mechanism, but I know that it impacts a lot of important other pathways and cellular functions. It reduces cardiovascular inflammation, influences other key mechanisms related to longevity. And what was known from this Honolulu Heart Program with FOXO3 is that if you have the most active version of the gene, the highest expressing form, that you were 3 times more likely to live to 100 healthy. And this has been replicated in other populations and other studies thereafter. And so to your point, FOXO3 is one of the most important anti-aging or longevity genes, if you will, that is currently known. And so going back to the worms, to the
I have a question about
elegans.
this. How many copies do we
Okay.
have? Do we have just one copy of the FOXO gene in human genome, or just multiple of these?
Yes, you'll want to have a GG allele, to have the most active form, and so there can be, the GG or other forms, and so that's what we're looking at.
So just
But
one gene, different forms.
yes,
Okay.
yes, and that's my understanding. And so going back to the worms, they have a gene called DAF16 that is essentially their version of the FOXO3 gene. And the hypothesis was if you knock out that gene, would you still have the lifespan benefit that was found in the C. elegans? And in fact, when you knocked out that gene, essentially got rid of the FOXO3 gene in in the worms, then the lifespan benefit went away. And so that contributed to the understanding that most likely astaxanthin, at least in the C. elegans worm model, was acting through essentially the FOXO3 pathway.
Yeah.
So based on that,
This is a study that you have
understanding-
done. It was done with your
this was
astaxanthin?
a published. No, this was just with other astaxanthin on the market that was sourced, from a laboratory source or from another commercial source, and done by other researchers in the international research community. So it's based on published research that we and others, would take to inform us with our, future research. And so we connected with our collaborators at the University of Hawaii who had this model in mice looking at FOXO3 activation. And we thought, let's see if astaxanthin potentially can activate or increase the expression of FOXO3 in the mice. So our researchers at the University of Hawaii are looking at everything related to longevity, and based on the fact that that astaxanthin appeared to work through the FOXO3 pathway or the DAF16 pathway in the worms. We wanted to just see, well, let's see what astaxanthin does with FOXO3 in mammals. So that was essentially the idea for, you have this model looking at FOXO3. We know that astaxanthin appears to act through FOXO3 in a basic model organism. Let's see what happens in the mice. And so we supplied astaxanthin to our collaborators at the University of Hawaii, and they found that the levels of FOXO3 were increased specifically in the heart tissue. And so that was the first time that anything was shown to improve or increase FOXO3 activation in mice with astaxanthin. And so based on that research finding, which built on the prior longevity research with astaxanthin, our researchers at the University of Hawaii went to the ITP and proposed that they test astaxanthin, because the way the ITP works is each year they'll take proposals for longevity candidates to test, and they'll get a lot of proposals that all hopefully have some merit, but then they have their committee and they'll select the lucky 5 or so agents each year to start in that year's cohort. And so each year they'll take a few thousand of these mice, split them up into groups with control and the various treatment groups, and then follow them throughout their life spans. And so in our case, our collaborators at the University of Hawaii went to the ITP and said, hey, we think astaxanthin is a longevity, molecule of interest. Look at our data here in mammals with FOXO3 activation that builds on the other data and the other mechanistic understandings. Let's see what happens. And the ITP agreed that would be worth studying, and so they asked us to then go ahead and work with them on supplying the material. And so we not only supplied the ITP with our form of astaxanthin, but we also helped them with the dosing. They did a pilot study before the main study to make sure that they could incorporate the astaxanthin into the chow or the feed for the mice, and then make sure they could get appropriate blood levels. And so we were able to get some preliminary PK data, pharmacokinetic data, or at least absorption data with the mice, and then ultimately continued to supply the astaxanthin throughout the multi-year study, which started in 2019 and ended in 2023 and was published in December 2023.
Wrote in the study that also the astaxanthin has a very good safety profile. People ask me in my community about the safety profile, especially when they use pretty high doses in the study. So let's speak about the safety profile. What are the possible side effects that people may experience? I intuitively thought about pink skin, although I read a study where people have been taking 40 milligrams of astaxanthin for like 4 weeks. There was no impact on the skin, but we do know that it is being accumulated in our bodies. But having pink skin, I don't view that as such a huge problem. I mean, a lot of people, they just I mean, if it's sexy enough for the salmon to be, then maybe it's good for me as well. I'm joking.
Yeah, and the flamingos. Yeah, exactly. And so I'll just touch on, we did a study in cardiovascular patients in humans that was 12 weeks of dosing, and it was a randomized placebo-controlled trial that explored low dose and high dose. The high dose was 96 milligrams, so 8 of our 12 milligram capsules, and we did not see any skin pigmentation, in that over that 12-week period. And then there was an open label extension where people could continue to take 24 milligrams, 2 capsules a day, for the remainder of a year. And actually, because this study was conducted or started just a year or so prior to the pandemic, it essentially went on hold during the pandemic, but people continued to get the open label product, and continued on for much longer than a year. And at that tail or open label extension of the 24 milligrams a day, again, there were no reports of turning pink or anything like that. So I don't think that's a concern. I know with other carotenoids like beta carotene, if you eat too much, your palms could turn orange, for example. And so we haven't seen that with astaxanthin. I'm sure that at a high enough dose, if you did that consistently, there could be some skin pigmentation. But I think at the doses that are typically used, that's certainly not an issue. But in terms of the safety, there was a very robust set of toxicity studies that were done many years ago, actually before the FDA approved astaxanthin to be incorporated into animal feed, because it's a really important component of feed for salmon. Like farm-raised salmon are actually fed astaxanthin in their diet. They're not dyed pink to some people believe they're artificially dyed pink.
But it's not your astaxanthin, right? It's like a synthetic form. It's not your astaxanthin.
It's not our astaxanthin. It is a synthetic form.
It's not tell us the difference. Like when they buy wild salmon versus farmed salmon, what type of astaxanthin and how do they get, and how does that affect the longevity benefits potentially?
Yeah, so the wild salmon will contain astaxanthin that is naturally produced by microalgae, and then it works its way up the food chain into the salmon. And if you look at the different types of salmon, the deeper the red or the pink, the more astaxanthin it contains. And the highest levels per serving may be 6 or 8 milligrams in a meal of salmon. The farm-raised salmon will contain astaxanthin that is of synthetic origin, but incorporated into their feed. and probably has lower levels just because the salmon farmers need to only include enough for the salmon to be colored pink, but it also importantly helps with their health and vitality, their growth and development. So it's not just helping them with their color, which is great for marketing, a piece of salmon, but also helps the salmon to stay healthy as they're growing. Okay, so to clarify, so in the wild, microalgae produce astaxanthin as a defense mechanism against UV light from the sun, or other phytoplankton, and it'll work its way up the food chain, and that's how salmon will consume it. But for the natural, quote, natural forms of astaxanthin that are on the market in dietary supplement form, those are typically extracted from microalgae that is farmed. And so people will grow the microalgae in man-made systems, but it's still the natural microalgae that you would find in the wild. And so you'll have different types of systems to grow the microalgae. 25 years ago, I worked in Kona on the Big Island of Hawaii, in the ponds that grow the microalgae that produces astaxanthin. And so I worked in the production there. And we launched one of the first 2 dietary supplements on the market back in that 2000 timeframe, 1999, 2000. Doing that type of production, though, especially the open pond production, is prone to contamination because you're exposed to the elements. You have the volcano nearby, so there's VOG. You have the airport nearby, so there's jet fuel. You have birds flying over. You're outdoors, and so there are those potential risks. It could be entirely closed tubes where you grow the microalgae. There are groups in, for example, Washington State in the United States and in Iceland that have astaxanthin production systems, or microalgal, Haematococcus pluvialis is the particular type of microalgae that is very good at producing astaxanthin. And so you can either grow it in these open ponds, like on the Big Island of Hawaii, where you have these big ponds full of water and the algae that grows, and it starts out light green, and then the sunlight hits it and it turns bright red as the astaxanthin is produced. And then you extract out as much of the astaxanthin as possible from the algae. You can also do it, like I mentioned, entirely in closed tubes that would be less prone to contamination. But in either case, you are producing the microalgae and then extracting the astaxanthin from that algae. But you're ending up with about 5 or 10% astaxanthin in that extract from the algae. And then the other 90 to 95% is other matter, content from the algae, which is not a bad thing necessarily. It's just not the active astaxanthin, that you're looking for. And so going back to my origins in this 25 years ago or so, after doing the microalgal production of astaxanthin, we thought, hey, this actually could potentially have pharmaceutical applications, given its intriguing impacts on oxidative stress and inflammation. And but if you want to develop something as a pharmaceutical prescription form, it has to be highly, highly pure and consistent, and that is pretty hard to do with a extract, from an algae or a plant. So we partnered with some of the European manufacturers of synthetic astaxanthin and worked with them to develop a pharmaceutical form of astaxanthin. And that was much more pure because you got just the astaxanthin molecule. And then you could either esterify it or formulate it to make it bioavailable. And so we worked on both sides.
Is any different? So people out there are going to ask, what am I buying? When somebody buys astaxanthin over the counter, do they get what you mentioned? What you mentioned is a higher purity. They get more of the active ingredients, because I know there are different isomers as well, different types.
Correct. Yes. So most of the astaxanthin supplements on the market are the microalgal astaxanthin. And in the microalgae, the astaxanthin has the SS isomer, which is, if you think of isomers as like hands, the end of the molecule can have either 2 left hands on either, a left hand and a left hand on either end of the molecule, or 2 right hands, or a left and a right hand. And in the microalgae, it's the SS form, the kind of 2 left hands, if you will. But in shrimp or other crustaceans, you'll find the RR, the 2 kind of right-handed forms, or the meso form, which is a left and a right-handed form of the optical isomers. But we don't have longevity data on that in humans, right? On this isomer that in shrimps, for example, we only have the
all the longevity data we have is on this SS isomer, correct?
No, not necessarily correct, because the ITP program was conducted with our form of astaxanthin, which is produced by natural product total synthesis, and that is a racemic mixture of the SS, the RR, and the meso, and so that was demonstrated in the ITP to extend lifespan.
It's three types. It's a mixture of 3 types of astaxanthin in essence.
Of the 3 isomers, it's 3 or 4 depending on how you think about it. It's the left, the SS, it's the right, the RR, and then you have the SR and the RS, which are identical. So whether you look at it as a 1 to 2 to 1 ratio of SS, meso, and RR, or a 1 to 1 to 1 to 1 ratio of SS, SR, RS, and RR. So essentially, it's an even mixture of all the optical isomers.
So would that be correct that our thinking about astaxanthin is very similar to vitamin E, that you have different types of tocopherols, gamma, alpha, whatever, so all the same. So when we speak about astaxanthin, I think it helps for the viewer to think, to be more smart, to understand what exactly they are buying. So when they are buying your product, they kind of get full spectrum astaxanthin with the 3 types, correct?
That is correct. And in some cases with other molecules, the isomeric form has a significant difference in safety or biological activity or how it binds in a cell. It can have a very important difference. With astaxanthin, it doesn't appear to have any difference. From a safety standpoint, the safety of the mixture of the isomers or individual isomer like SS both have been tested. In fact, the most rigorous toxicity testing has been done with the racemic mixture, which supports the safety of all the isomers. From an efficacy standpoint, a lot of the basic research, for example, that's done in the model organisms, or a lot of the animal studies have been done with synthetic astaxanthin, which is typically the racemic mixture of the isomers. The ITP study that we supplied the material to was the synthetic racemic mixture of isomers. A lot of the human studies, though, have utilized the microalgal astaxanthin, which is the single SS isomer. And so a lot of the human data is with that. But I think that there is one study out there that was published in 2013 that was commissioned by the head of marketing for one of the microalgal astaxanthin companies, who is not a scientist, that looked in vitro at antioxidant performance. And in that particular study, which I don't think was designed properly to get the same concentration of the astaxanthin into the system from both natural and synthetic origins, that study showed that the natural performed better. But for example, we have done studies looking at antioxidant performance with each individual isomer, extracted, separated out and tested individually, and also the mixture of the isomers. And the superoxide anion scavenging was basically the same across all the isomers or the mixture. And that was a well-designed study that demonstrated that, again, the isomers did not appear to have a difference. As a side note, we have synthesized single isomer astaxanthin, just the SS, like exactly as you would get from the microalgae. And we've also tested that in animal proof of concept studies in the past, and that worked well too. So I don't think the isomeric form makes a biological difference. And so I think that's kind of some misinformation that's been put out there from a marketing standpoint by some of the microalgal astaxanthin companies that either the SS form is better than the other forms, or that the microalgal, quote, natural form is better than synthetic. I think the microalgal and the synthetic forms have both been demonstrated to be safe and efficacious. And so if you can get the astaxanthin molecule to the cell, it's going to be functioning the same, I would believe. It's what's different is how you produce it. What's the purity, what's the consistency batch to batch, what's the formulation, how well is it absorbed? Also, just are you actually including the appropriate amount in the capsule in the bottom? Because I know that there are, there's some, there was a study that was published showing different brands of astaxanthin, and they measured the amount of astaxanthin in the capsule, compared it to the actual label claim of 12 milligrams versus actual, and a lot of the brands on the market were under filled. They had an underage, and the amount of astaxanthin was less than the label claim. So there can be those types of differences. But if you assume that you adjust for getting the same amount of astaxanthin into the tissues, into the cells, I think whether it came from a natural origin or a synthetic origin, the molecule is the molecule. It's the same carbon, hydrogens, and oxygens, and the isomers don't appear to have a difference. So we think of astaxanthin as astaxanthin, but then you back up from a feasibility standpoint and figure out, okay, what's the best way to make it and deliver it to people? And again, working in both, I think both have merit.
So let's. I'm going to create a big picture of what the consumer can get, and correct me if I'm wrong. So. When people consume astaxanthin, they can get it from wild salmon, for example, farmed salmon. From farmed salmon, they're going to get the synthetic form, which includes the three isomers, right? The SS, RS, and RR. Correct? They're going to get all three of them. When they when they consume the wild fish, wild salmon, they're going to get mainly the one with the SS, right? Because I guess. I'm sorry, because it also consumes shrimps. So you're going to get both the RR and the SS, probably.
It's mainly the SS. I believe there's some of the meso or RR, but I think it's predominantly SS, if I recall correctly. But in crustaceans, shrimp, crab, lobster, you'll get more of the RR and meso forms.
And then they can buy a supplement. I'm not speaking about your specific brand, but the common ones, it's usually the SS. The natural one is usually the SS when they buy, correct?
Correct.
And in your specific brand, they get a mixture, or they get also the SS?
In our brand, it's the mixture. It's the racemic mixture. My side note earlier was that we have synthesized the single isomer and tested that, and that also worked really well. But we did not find a difference between single isomer versus the mixture of the isomers. And it's easier to produce the mixture. And actually, the most rigorous toxicity testing was done with the mixture as well. And so that's why we utilize that form for the consumer product. But we do have future plans to also launch a microalgal form to expand our product line, just so that people have choice if they prefer one form over the other. We think that scientifically, the synthetic form has the strongest evidence for longevity with the ITP, with safety, with the toxicity studies, and certainly bioavailability, like we tested in humans, and manufacturing purity that doctors and other health-conscious consumers should find important, knowing that it's pure and consistent batch to batch, capsule to capsule. So we think the synthetic form that we refer to as BioPure for our form of astaxanthin is optimal. But at the end of the day, the world just needs to know about astaxanthin. A lot of people don't even know about it. And so whether they get it from a microalgal astaxanthin supplement or a synthetic astaxanthin supplement, I think all of that is a win. Just the more people that take astaxanthin, the world will be a healthier place. So that's kind of what we're supporters of in general.
And in the FOXO experiment you have done with 90% increase in the gene expression in the heart, it was used exact one in the ITP, correct? They used the mixture of the three.
So the form in the FOXO3 study was our synthetic astaxanthin, but in that case, it was one of our pharmaceutical candidates that was in a esterified form, I believe, of the SS astaxanthin. Because for the pharmaceutical purposes, from an FDA regulatory standpoint, it's easier to develop a single isomer because the FDA will require you to show safety, absorption, other things with the different isomers. And so it's even if it biologically doesn't make a difference, you have to prove that very extensively with the FDA. And so we actually had programs in place looking at all the different isomers, and it's not really feasible to do. So we did a single isomer synthetic version for a pharmaceutical development purpose. And so we had material on hand that we utilized for the FOXO3 study. But whether you have
it proves your point.
Form,
it proves your point in essence that it doesn't matter. I mean, it activates longevity. And this is the good time to go to the dose. My interpretation when I look at the ITP, it uses something along the. I mean, I've seen they wanted to put higher dose of astaxanthin, but they only managed to get like 46%, which is about 1,300 milligrams per 70 kilo human. Am I right on this ballpark? So if people want to know how much astaxanthin the ITP gave to the mice, am I correct when I'm thinking about like 1,300 per a male like myself?
I think a better way to look at it is correlating blood levels of the astaxanthin, because
Okay.
when you look at the dosage used in the ITP, you'll find that's reported as parts per million in the chow, in the feed. And
Yes.
that was based on trying to mimic the doses that we utilized in our animal proof of concept studies, which was often in the 500 milligram per kilogram range. And that was where we showed the most benefit in the animal studies. And so that was essentially the reason for the dosage that was used in the ITP. To your point, when they actually did dose analysis of the chow to figure out how much astaxanthin was in the chow, there was some variability in the results that, as reported in the paper. And what we don't know is, were there actual variations in the amount of astaxanthin in the chow batch to batch when they made it? Or was it something where their analytical methods were not optimal for the measurement? Because astaxanthin is actually pretty tricky from an analytical chemistry standpoint, to make sure you have consistent, reliable methods. And so there's a bit of an unknown there. But we certainly know that based on the pilot feeding study, that the blood levels were in the several hundred nanograms per milliliter in the plasma. And when we look at human studies that we've conducted, we did a human pharmacokinetic study looking at microalgal astaxanthin, one of the leading brands on the market, and then our form. And I can discuss that study in more detail in a moment, but for this purpose, we had a maximum concentration of several hundred nanograms per mil. I think it was 480 nanograms per mil, in that following a single dose of 24 milligrams, two 12 milligram capsules of our astaxanthin product. And so,
but I would be the devil's advocate and going to say astaxanthin has been accumulated. Why would we care so much about blood levels? I mean, we don't. It's being accumulated in fatty tissues, in the brain, in the eyes, in the membranes of the skin.
Yeah.
And why would blood levels be such a good indicator?
Well, it's a surrogate. I mean, ideally, you would have all the tissue levels in humans. We can't get tissue levels easily, and so looking at the absorption over 24 hours and the half-life can give you an idea of how well it's absorbed, and then what type of dosing regimen is optimal. In animals, you can look at plasma levels and you can look at tissue levels, and we have done that in the past, and so. I think that the best that we have is looking at the plasma levels in humans and the plasma levels in the mice in this study, and trying to see if we're in the ballpark. And that's at this point the best that we would have. And so based on our human PK study, you would conclude that probably 1 to 2 12 milligram capsules of our form of astaxanthin, which is more bioavailable than other forms, but 1 to 2 12 milligram capsules of our AX3 astaxanthin would get you in the range of what the ITP observed in the mice in the pilot study prior to the main ITP multi-year study. So that's probably the best idea of a correlating dose.
So your conversion, what they used in humans, just about one capsule of highly absorbable 12 milligrams. This is it, nothing else.
Yes, 1 to 2. And then if you try to do it the other way, where you look at the actual, parts per million or milligrams per kilogram used in the chow, or actually ingested by the animals, there's several conversion factors you would have to factor in. There's the typical body surface area conversion that is used by the FDA. Yeah, and so based on rodents to humans or canines to humans, there's different conversion factors. But in addition to that, carotenoids are not absorbed as well in rodents compared to humans. And so there's additional conversion factor that has to be accounted for. And so, because otherwise, you calculate much higher levels.
Lack of lipases or whatever?
Yeah, I don't think we know the answer to that question. It
that's
could
very
be
important.
that they don't have...
what you say, let me stop you there, because what you said is super important. It means that the conversion, we should not use the regular academical conversion here.
Yeah, correct. Yeah, because that would lead to very
this is pretty shocking.
incorrect dosing. Mm-hmm.
Yeah, because if I do the regular conversion, I get to like 1200 milligrams, and you say it's like it's a 100-fold less.
That'd be... Yeah. No, it's very hard to get astaxanthin into rodents, and so
I see.
Yeah, it's way less efficient.
That's great
And
news
that
for
could
people
be,
if they want to imitate the
is, yeah,
results.
yeah.
It's a great news.
Yeah, it's not great for our mice pets, for example. That we want to extend their lives. But I mean, you can feed them enough and get similar blood levels. It's just much more on milligram per kilogram of body weight basis. And so yeah, to your point.
So the blood. So just to verify, so the blood is basically a way to see how much actually gets into the system. That's why you feel it's a better measurement as opposed to how much to consume, which is absorb less. Okay.
Yes, yeah. And so just because you consume it through food or, a capsule form, tablet form, doesn't mean that it's all getting absorbed into your blood, into your tissues. And so looking at the blood is much better than just trying to calculate what was the dose up front. And so, that's a much better way. But to your point about the absorption, in humans, it might be that there are particular scavenger receptors, protein transporters that are, present in the humans that more effectively absorb carotenoids. And even within humans, there are inter-individual variations, in genes that encode for certain scavenger receptors in the enterocytes that may promote the absorption of carotenoids in addition to passive diffusion. And so that's an emerging area of research for carotenoids in general, not just for astaxanthin. And so that it'd be really great to know more, but it's probably, I would hypothesize, related to that, where the mice maybe from an evolutionary standpoint didn't evolve in a way that promoted their carotenoid absorption, unfortunately. So sometimes they'll even find intravenous or other forms of administration used in rodent models to get it in there for testing purposes.
I guess they don't have the history of going to fish. They just unlike us.
Yeah, exactly. So perhaps
we inherently lived near the sea most of the time.
Yeah. And so from a longevity standpoint, from a basic health and potentially increasing lifespan standpoint, the best we would know from the ITP data and trying to extrapolate or translate that to humans would be 1 to 2 12 milligram capsules of our highly bioavailable form of astaxanthin. But with that said, for example, we did a human study, like I mentioned, in cardiovascular subjects that had a placebo dose and then a 24 milligram 2 capsule dose as a low and a 8 capsule 96 milligram high dose. And we found that at the 96 milligram 8 capsule dose over 12 weeks, that there were improvements in terms of reductions of cholesterol, but importantly, LDL and total cholesterol, but importantly, oxidized LDL, which we sent to a special lab to analyze for the oxidized LDL, and we saw reductions.
When you compare the dose, when you compare you saw better improvement with 96 versus the 12, you said? 96
versus 24.
24. Yeah. So this is already 2 capsules, and you see even improvement with about 8 capsules.
Yeah, so there is a dose response, and that is consistent with animal studies that show dose response with increasing benefits at higher doses.
Also in the mortality studies in the fruit flies and the worms as well, the higher dose perform better in longevity, from what I've read.
Yeah, and given astaxanthin's exceptional safety, it doesn't appear to have dose-limiting toxicity. This is something that people can experiment with higher doses, but obviously they should work with their doctors or healthcare professionals, and if they're on other medications or supplements or have other disorders, that should all be managed. But generally speaking, astaxanthin has been shown to be exceptionally safe. And just to wrap up on the toxicity studies, the FDA, before they allowed astaxanthin from any source, but it was synthetic that was ultimately used for the salmon feed, for example, before the FDA would approve the use of astaxanthin, technically as a color additive is what it's classified as from a regulatory standpoint, even though it contributes to their health as well. Before the FDA approved that, there had to be this very extensive, robust battery of toxicity studies. So it included single dose toxicity at like thousands of milligrams per kilogram of body weight, which had no adverse findings. There was subchronic.
How long did you?
Well, that was a single dose, in multiple species, and then there was subchronic, which is like 3 months of dosing in multiple species at probably a couple thousand milligrams per kilogram of body weight. Which, for comparison to humans, if we took 6 capsules a day and 72 milligrams of astaxanthin, that would be effectively 1 milligram per kilogram of body weight. And most people are taking, a sixth of that or a third of that. And so we're taking typically fractions of 1 milligram per kilogram of body weight. These toxicity studies, the single dose, the subchronic, were at thousands of milligrams per kilogram. But like we talked about before, the absorption in not just rodents but other animals are not as good. It's not as good as humans. But with that said, there were also longer term studies, 1 and 2 year carcinogenicity studies at very high doses with no negative findings or with no adverse findings. There were genotoxicity studies looking at DNA damage, nothing found there, and developmental and reproductive toxicity studies looking at mothers and offspring, no issues found there. The only finding of any interest from all of these toxicity studies was in a female rat. There was a benign liver lesion that is not clinically relevant or translatable to humans. And that was the only thing. But if you look at in Europe at the EFSA findings or report related to astaxanthin, they singled out that one finding from the female rats, which was, again, not clinically relevant to humans, but they used that as a dose-limiting toxicity and then took the dose tested lower than that and said, okay, that's the safe dose of astaxanthin. So in Europe, the quote safe dose of astaxanthin is minimal. It's like a couple milligrams a day or something, which is I think not the appropriate conclusion. So
they're limiting the dose you can sell in Europe. I live in
not
Europe.
that you can sell, but what they recommend as what they think is the limit for known safety. But I think that's a very extreme conclusion drawn from a single, like I said, female rat and with a particular type of liver lesion that is not clinically relevant to humans. And if you take that aside, and look at all the other studies looking at safety, toxicity, there were no issues. And I think that's also confirmed with the 100 or so human studies done with astaxanthin that have not found any side effects of clinical significance. So
I do have a question about that. Yeah. So in my community, they ask me, they know that all of these fat-soluble antioxidants, they tend to interfere, especially from land plants as opposed to algae, which is so ocean plant. So they know they interfere with the production of free radicals, which are essential for the benefits from exercise. Have you seen? So I have seen the exercise studies I have read. They showed that for people who over-exercise, it reduces the damage, it reduces chronic inflammation. I read multiple, in Serbia, there was a bunch in Serbia, they're very interested in that. But I haven't seen a study that tried to see if there is a negative impact on antioxidant activation by exercise. In other words, does astaxanthin is going to offset some of the antioxidant activation coming from free radicals during the intense exercise? Have you seen anything about that?
I have not seen anything about that. It would be great to explore that further. But you can look at things like in nature, salmon obviously consume a lot of astaxanthin, and they have to go through a very strenuous, long journey upstream to reproduce. And so that long swim is something that if astaxanthin was negatively impacting these things, you would suspect that maybe it would hinder their ability to have that miracle, feats of strength and endurance to swim upstream. So there is that. And then in humans, if you look at various studies related to exercise and performance, there are varying results, which I think are probably based on study design and dosing. But if you look at muscle development, or in particular, there was a study in the elderly looking at sarcopenia, which is muscle deterioration, and they found that astaxanthin supplementation supported increased muscle size and strength. And so that, I think, is an important finding to show that
17 humans, 70?
It was older adults, elderly. Yeah, I don't recall the exact age, but it's probably more than 60. I would guess 60, 70, 80, in that age range. But certainly in older adult population was where that was demonstrated. I don't know of research looking at the same in younger adults or middle-aged adults. And so I think that's an area that would be good to explore further. But A lot of athletes, triathletes, for example, have historically used astaxanthin, and so I would think that if there were a major issue with its interfering, with recovery or with muscle, growth, that it wouldn't be so, well utilized in the athletic community.
Yeah, at this point, I would agree. I think because they showed, for example, European footballers, people who play football professionally, they basically over-exercise, and astaxanthin, in essence, reduce HSCRP, so it reduces systemic inflammation. This and HSCRP also controlled by NRF2. So what it tells me that astaxanthin did not offset the activation of NRF2 by exercise; it just enhanced it. Otherwise, I wouldn't. Otherwise, I would not have seen this impact with reduction of HSCRP. You know what I'm talking about? The studies I speak about. So
yes, I've seen. There were studies in the young soccer players, as we would say in the U.S. European footballers, and we've seen reductions in HSCRP. For instance, in our cardiovascular clinical trial, when we looked at the diabetic subpopulation, there was reductions in HSCRP as well, and it's known to activate NRF2. Like you said, NRF2, and so which has an important role in antioxidant, anti-inflammatory, and other cellular functions. I think it most likely doesn't have a negative effect on any type of things related to exercise. If anything, it helps you with endurance and recovery. But even though there are more than 3,000 papers with astaxanthin and 100 or so human clinical studies, there's just so much more that is left to learn and to be discovered. So I think additional research in this area would be great to see.
Yes, and you spoke about people asking about the dose. So I try to pinpoint the dose as much as possible, because to me the dose really matters here. I mean, to me, I think there is big difference in longevity activation between 2 to 4 milligrams to 12, 24, 36, somewhere there. I think I think that there is dose dependency from what I've seen here. I'm pretty certain about it. In the 96 versus 24 study that you have seen, did, besides effect on cholesterol, did you measure anything else? Maybe HSCRP, anything that could because to me cholesterol isn't very good metric for longevity. I mean, it doesn't tell me what happens in the DNA level, but HSCRP, I can say maybe NRF2 was activated. Anything that you have seen, those different impact on the dose.
Yeah, so the HSCRP was reduced in the diabetic subgroup in that study, and in the overall population, it was reduced, but it didn't quite hit statistical significance because I think the study wasn't quite big enough. And unfortunately, with HSCRP, you can have some variability in measurements, in individuals. And so I think a larger study would have most likely demonstrated a reduction of HSCRP, but other studies in humans have demonstrated HSCRP reduction as well. But in that study, it wasn't just cholesterol. Like I said, it was also oxidized LDL. And so oxidized LDL is a really bad form that can contribute to heart issues, and which, the leading cause of death is, heart disease. If you can, even though this is a supplement and you can't claim it prevents or treats heart disease, maintaining your heart health is really important. And so if you can reduce the the oxidation of the LDL, and you can reduce inflammation systemically, those are things that you would conclude should hopefully extend lifespan and health span.
So it was better result with 96 milligrams versus 24 in the oxidized LDL?
Correct. It was, yeah.
Was
and the
the
HSCRP reduction was
significant?
Similar.
The p-value was good?
It was, yeah. The p-value was only statistically significant actually in the high dose, and it was like a 10% reduction in the high dose. And the CRP reduction was about 30% reduction in both doses, although it didn't, like I said, hit statistical significance. Again, with a larger study size, or perhaps modification of the inclusion and exclusion criteria to get a more similar patient population, you probably could have demonstrated statistical significance there. But like I said, there's other studies showing reduction of HSCRP in humans. But yeah, so the best results in terms of the LDL cholesterol reduction, the total cholesterol reduction, the oxidized LDL, and even blood pressure was all in the higher dose. And that is consistent with when we look at the blood levels seen in our animal studies that look at various aspects of heart health and liver health. The blood levels seen there were similar to what we measured at that high dose in humans, which, like I mentioned before, that 24 milligram single dose, we got around 480 nanograms per mil after a single dose. And then in the cardiovascular study, when we looked at after 3 months of dosing, we were curious what the blood levels were, and it was around 800 nanograms per mil in the 24 milligram daily dosing for 12 weeks, and more than 2,000 to 2,500 nanograms per mil in the higher dose, 96 milligram dose over 12 weeks. And so again, that's not measuring tissue, but it's measuring the plasma as a surrogate, and showing increase. It's not a linear increase of astaxanthin, but it's certainly
reduced. The blood levels reduced, yeah. And do you think dividing the dose? Do you think dividing the dose? For example, coenzyme Q10. If you divide the dose, I know with my clients, they're gonna get better coenzyme Q10 blood levels. Do you think in astaxanthin there is a limitation to the absorption of humans per one dose? So we need to separate the dose.
Perhaps we haven't tested that. I imagine you could overwhelm the system with too much at once, potentially. But
especially
I think
the mice. Especially if the mice have limitation. Why would we not have some limitation at some point?
Mm-hmm. Yeah, exactly. And so that again would be interesting to explore. Our studies have been either single dose or twice a day dosing, morning and evening, and that was for multiple considerations. For that cardiovascular study, we did morning and evening dosing to split the dose. So rather than taking 8 capsules at once, you would just do 4 in the morning, 4 at night. It just makes it a little bit easier to do. And like you said, potentially splitting it up may have give your gut, a better chance to absorb as much as possible. But we haven't studied side by side or head to head, like 1 capsule morning and night versus 2 capsules at once, or 4 capsules morning and night versus 8 at once. That'd be interesting to explore.
Just to see blood levels, to see the impact on blood levels would be very interesting.
Yeah. It would,
like 96,
but it is
maybe like 24, or 48 at once. That would be interesting one.
Yeah, exactly. And but from a half-life standpoint, so if you're looking at the pharmacokinetics, typically if you have something that has a 24-hour half-life, then that is conducive to once daily dosing. And so when you look at it from a pharmacokinetic standpoint, it's perfectly fine to take once a day. That's kind of an optimal dosing for an agent that has approximately a 24-hour half-life. That's good for daily dosing.
So when you say half-life, you speak about half-life in the blood?
Yes, in the blood. Yeah, because again, you can't get multiple samples from tissue in the same animal or human.
It tells me that astaxanthin doesn't just go and accumulate immediately in the brain. It just hang on in the blood. It takes some time to become integrated into the membranes, right? It's not immediate integration. We're talking about a process.
It goes through a process, and so it has to go through the gut, and it'll get packaged into mixed micelles, with bile salt and lipids, and then absorbed across through the enterocytes, and then it'll get packaged with lipoproteins and distributed throughout the body. And so it protects those lipoproteins like LDL from oxidation, but then uses those to be distributed to the major organs. And so it takes time to get to the organs, and then and if you're taking it every day, then it hopefully continues to replenish the tissues.
Be very interesting to see the impact on the way you described it, because it's being carried by the lipoproteins. What would be the impact on the plaque formation? Because we know plaque formation begins with the accumulation of the ApoB molecules, but maybe if astaxanthin is going to be there, and the immune system comes, maybe the reaction of the immune system to the accumulation of the ApoB molecules that are being stuck in the blood vessels, maybe it's going to be less violent. Who knows?
Yeah, and so again, with the caveat that we can't promote astaxanthin as a drug to prevent or treat disease, there's a lot of interesting research, for example, in animals looking at cardiovascular applications. And so, for instance, we did a study with a collaborator at the University of California, San Diego, who had a model looking at atherosclerosis or plaque buildup in the arteries of mice. And these mice were genetically predisposed to accumulate plaque in their arteries. And so we looked at placebo, low dose and high dose, and we demonstrated through imaging a reduction of the plaque in the aortic arch of the mice, a very significant reduction. So to your point, yeah, I mean, I think we would see reductions in the plaque, which is really important. It also impacts platelet aggregation. We had multiple studies in both rodents and canines showing prevention of primary and secondary occlusion or thrombosis, in the arteries.
Let me add to the audience. My wife, she had a stroke, and platelet aggregation basically causes the clot to be formed in the first place, and she had a clot going to her brain. So the platelet
so sorry to hear that.
So the platelet aggregation, just for the audience to understand, really. So the atherosclerosis reduces the space for blood to go in, but the clot comes and give the final blow to the whole situation. So
and if it's all inflamed and oxidized, it's more likely, to have, a soft plaque, create a clot. So it's something that you definitely want to reduce the oxidative stress in the environment, but also reduce the plaque buildup, itself and the platelet aggregation.
By the way, any studies are gonna send me afterwards. I can put in the description for people to look at themselves. And just to close the dose aspect of it, for me, I began to suspect about the higher dose does a better job because of the longevity studies I mentioned, and the fruit flies and the worms. Any indication that led you maybe to suspect that higher, besides the 96 versus 24 milligram study in humans, any indication that led you to believe that maybe we need to go higher than 4 milligrams, 12 milligrams? Anything that gives you this hint?
Oh yeah, so multiple things. So of course there's hundreds of animal studies and in vitro studies, all, or I would say many, showing a dose dependent response. And so higher dosing, it would be thought to have better results. And so there's just lots of studies supporting that. But then also just anecdotally, so not scientifically, but just feedback from our community or just the community of people taking astaxanthin, or even just people in my family or friends. My father, or he plays tennis very often, and he's found that higher doses, and he's in his mid 70s and plays singles hard court tennis multiple times a week, and he's found that higher doses have really helped him with his joint mobility. And same thing for his tennis partners, and they're really pushing the envelope.
What is how the high doses?
The power
when he said higher doses helped him, just what are you speaking about?
He's experimented at a range of doses, and so he has worked even as high, and we wouldn't necessarily recommend this because it's not practical and not widely studied at this level, but he is taking, and his buddies are taking on the order of 16 capsules a day of our product, so double the high dose in our cardiovascular study, and finding increasing benefits there. And so that's where it just supports that higher dosing is typically going to have, hopefully should have a better result. But again, and I think that is covered from a safety standpoint with all the toxicity studies, but it's not something that we would promote or recommend that everyone take. But I think the important takeaway is that you can start at the 1 or 2 capsules a day of 12 milligrams of our highly bioavailable form, and then you can give yourself a few months, and you can do self-assessment of how your joints and muscles feel, or cognitive function, or skin, or eyes, energy levels. You can assess those things, sleep, etc., and see how you're feeling. You can also measure biomarkers like we talked about, HSCRP, or cholesterol, or liver enzymes, or other important biomarkers of interest, and see, say after 12 weeks or so, if you're noticing improvements in areas that are of interest to you, or maintaining things that you really want to maintain. And then you can adjust your dose, because there is going to be variation in individual absorption. And this is something that's true for carotenoids in general. And so not everyone will absorb carotenoids or astaxanthin in particular the same. And so I think people ultimately, if they want to optimize, will have to titrate their dose. What would be cool to have is a consumer diagnostic to get the blood levels tested for astaxanthin, because I think if you could titrate to a blood level, that would probably be much better than a dosage level. We have methods to analyze astaxanthin in plasma, but that's at a private laboratory, and it's very expensive, and it's not conducive to consumer applications. But that is a future aspiration, because I think it would help people find an appropriate dose. But
is there evidence?
I think
is there evidence that all people, they absorb less? I mean, if we're going to speak about absorption as blood levels, do you think there is evidence? Maybe because the absorption seems a bit tricky. Do you think all people like A70, they need a bit more because they absorb less?
That's possible. I don't know of evidence to show that or not. There's been studies looking at ATP production in young and geriatric dogs, showing benefits, in both at the same dose. And so, but I don't know in humans. I think, again, you'd have to do a side-by-side test and see, or even follow someone and see if their levels actually decline with age. And so that's, at this point, we wouldn't know. But I think regardless of the case that you would have there, you'd want to look at your biomarkers or other self-assessments of health or longevity, and adjust your dose. And given the safety, there shouldn't be any issues of major toxicity, that you would typically find with other supplements or drugs. And so you can try a different dose, go up or down, and then adjust accordingly. The one caveat I'll mention regarding dosage as well is that it's not just the active ingredient, just the astaxanthin. There's also the other components of the formulation. The composition of the capsule, if it's suspended in oil, what type of oil, or in our case,
let's speak
it's
about
formulated.
that. Let's speak about the absorption, because your company uses a special formula. So I'm going to tell my very poor understanding, and correct me and tell me how you do it better. So my understanding is that astaxanthin is esterified, which means that we need the enzymes that digest the fat to be around to cut the link, and only then to be absorbed. But I've got the feeling this is not really what happens here. So can you explain, should it be taken with fat? And your formula, I mean, most companies, they sell it with some fat, some sunflower oil or olive oil. And in your formula, you use a mixture of carbs of some sort. And
correct.
So maybe you can explain the absorption of astaxanthin. Should it be taken with meal, without meal, and how your formula is different from what is being commonly sold on the market?
Sure. So the microalgal astaxanthin is typically esterified. And so if you look at the extract of the astaxanthin, the majority of it will be monoesterified. So it'll have astaxanthin esterified with a particular, a mixture of different fatty acids, but it'll be esterified with a particular fatty acid, and then you'll have some diesterified forms. So astaxanthin is esterified with fatty acids on both ends of the molecule, and a little bit of non-esterified astaxanthin, just astaxanthin, the molecule itself. But there's probably 10 or 15 different fatty acids that are esterified with astaxanthin, either mono or diesterified, in that extract from the algae. And to your point, in order to be absorbed, when it gets into the gut, you'll have to have the lipases, for example, you have to have the enzymes to cleave, hydrolyze, the esters, and essentially deliver the astaxanthin, for absorption, into the body. In the case of our pharmaceutical development, we esterified astaxanthin with amino acids that made the molecule very water dispersible. But again, the esters would be cleaved, and you would be delivering non-esterified astaxanthin to the bloodstream and to the tissues. In the case of our
that is gonna happen before the absorption, unlike chelated minerals,
it doesn't
where some of them, it's not chelated really. It just, yeah, okay.
No, it just helps for the dispersion in the gut. But then from there, it gets cleaved. And I guess at a high enough level, you might get a very small amount of the esterified form getting across, but it will still, at some point in the plasma or tissue, it will be cleaved and you would only have the non-esterified form. So it's not like you're delivering the esterified form to the cells.
But I think you have done something very useful, because it's very difficult to be dependent on the lipases, because we don't make a lot of them, and with the, I guess you need some kind of protease of some sort to cut the. So we have way more enzymes that are gonna cut the astaxanthin in the right time. Correct? I mean,
yeah, and so but with our supplement form, it's non-esterified. And so, but the thing is, if you just take astaxanthin non-esterified, synthesize it in the lab, highly pure crystalline astaxanthin, and you don't esterify it, it's not well absorbed at all. Whether you put it in oil or water, whatever, we've done studies and it's flatline, no astaxanthin detected in terms of it in the blood. It's not well absorbed. So you have to somehow either esterify it to, I think, allow for that dispersion in the gut and incorporation with the fats, or you have to formulate it in a way which, in our case, with our formulation, it utilizes, like you said, carbs. It utilizes a starchy matrix. And so you take the tightly packed crystalline astaxanthin that's kind of like a brick that your body can't really absorb, and you disperse it into a cloud or a mist, like a matrix of food starch, which if you think of a nice cloud of starch that astaxanthin can then stick to, there's also a little bit of glucose syrup in there, less than 1/10 of half a gram per capsule. So very, very minimal amounts that would be considered sugar-free from an FDA standpoint, but to help stick it together, and a little bit of cornstarch just to make sure that the, call it beadlet particles of the formulation don't clump and stick together, in the capsules. And so, but that formulation allows the astaxanthin to be water dispersible, which is a bit counterintuitive because you think of it as being fat soluble. But the hypothesis is that in your gut fluid, which is aqueous, in the GI fluid, if you can disperse the astaxanthin formulation uniformly or nicely in the GI fluid, then the normal process of incorporation of astaxanthin into the mixed micelles with the bile salts and the lipids, and then astaxanthin gets absorbed along with the fats. And so your body, it will best absorb astaxanthin with a meal containing fats. And so that's why
even
you want to
with your formulation. So
even with
ours,
your
product. So they always need to take it with meal anyway.
Yeah, and even if you have one with the oil suspension, like from the microalgae, the amount of oil in that capsule is pretty small, and that's and I think ideally, based on the research I've seen with carotenoids in general, like beta carotene and things, you'd want to have probably a few grams of fats, a bit more for your body to work with than just a small amount in a capsule. I mean, that would be better than nothing, but I think ideally you have a meal that has some fats in it, like a nice lunch or breakfast or dinner that has some fat content. And then following the meal, after your body is now starting to process those fats, you take the astaxanthin, in our case, the water dispersible formulation, which you can either swallow the capsule or capsules, or you can even open up the capsule and put it into water or a shake and it'll go right in, whichever way you prefer. But then it gets dispersed in the GI fluid and then absorbed the same way that fats are, and then brought into the body that way.
So let me summarize. Go ahead. Let me summarize what you said, and then tell me about the differences. So for the consumer, in essence, they have two options. They can buy the astaxanthin with oils. In that situation, they're going to get esterified astaxanthin. They would have to take it with fat. In your situation, we can say it's like evenly dispersed, non-esterified astaxanthin, right? It's like different, it's non-esterified, evenly dispersed astaxanthin. Now, a company that competes with you is going to say, okay, so what's the big deal? That we're going to get, why did you even bother to formulate it this way if we're going to get the absorption with the normal astaxanthin?
Yeah, so we did a head-to-head human study. So we took one of the leading brands of microalgal astaxanthin on the market, bought the bottle off the shelf, and got a group of human volunteers, healthy, normal-aged adults, and gave them 2 12-milligram capsules of the microalgal astaxanthin, so the esterified form in oil, and then collected their blood at multiple time points over 24 hours. Like 1 hour, 2 hours, 4 hours, 8, and onward throughout 24 hours. Sent those samples to the laboratory, and we have highly precise analytical methods that use like carbon-13 labeled astaxanthin that we specifically synthesize so that you can really quantify the exact amount of astaxanthin in the sample. And then we had those volunteers go home for a week and not take any astaxanthin supplement, of course, and also not consume foods like salmon containing high amounts of astaxanthin, and then come back a week later and then gave them 2 12-milligram capsules of our astaxanthin product, the non-esterified form in the starchy formulation, powder in a capsule. In both cases, they had a normal standard meal. And this was at a professional contract research organization. And again, collected their blood for 24 hours after administration of the 24 milligram 212 capsule dose. And when we compared the blood levels, we got 3 times the maximum concentration, the Cmax. Again, that was around 480 nanograms per mil with our form, and 150 or 60 nanograms per mil with the microalgal form. And then when we looked at the total exposure over 24 hours, which is the area under the curve of all of those time points, that was also approximately 3 times the absorption.
3 times.
Wow. 3 times the blood levels. But again, looking at Cmax as well as the maximum concentration, as well as the total exposure, the AUC over 24 hours, and the inter-individual variation, the levels that each of those 5 individuals had of astaxanthin was more variable in the microalgal group than with the with our form, the synthetic.
Less of a gamble. It's less of a gamble to the consumer.
But these were the same people. It's not just 2 groups of 5 people. It's the same people, and there was more consistent absorption in addition to 3 times the absorption.
That could also pose a risk for older population, I think. When they have such a variability, it could be that your product gives more confidence in the absorption. They're going to get it into the system.
And so we found that does give confidence to doctors when we talk to them, because we have our roots in pharmaceutical research for a decade, looking at astaxanthin as a pharmaceutical agent. And so, that's really our world is the science and communicating that. And so when we launched our consumer product, a few years back, we reached out to the physician community and the healthcare professional community and discussed these, properties of the formulation and the research results. And a lot of doctors are skeptical of supplements, because of the claims, the science, and manufacturing. And so this was something where we like to think of it as having somewhat the best of both worlds, where you have a very rigorous, production, kind of like a pharma-like production of a naturally occurring molecule. And so you're not giving an individual a molecule that was not present in nature that chronically inhibits or activates something that's not natural for the body. But this is a natural molecule that has been shown to be very safe and have a lot of important health and longevity benefits, but is being produced in a way that a healthcare professional or health conscious, consumer can have confidence in, that it's pure, it's consistent, it's well absorbed, it's consistently absorbed. So that's one thing that we found was, really powerful with the professional community.
How many people do you use in the absorption study? Do you remember?
The absorption study was 5 healthy individuals. So yeah, it's not the largest study, but it's randomly selected 5 healthy individuals. The cardiovascular study that I mentioned was about 40 to 60 people. We had an interim review at 40 subjects, and then continued to enroll and got up to 60 subjects.
The one with the plaque. The one with the plaque.
With the oxidized LDL and blood pressure and the plaque was an animal study. That was a mouse study.
Tell me. So the consumer gets probably 3x the absorption plus the confidence in the lower variability. So they know have more certainty they're going to get into their system. Very, very interesting. I have a question about the storage. A person buys the astaxanthin. Should they store it in the fridge? What would you recommend? Also, your brand compared to maybe other brands. Maybe your brand has different storage conditions.
Yeah, I think for the most part, most brands or ours are generally the same in terms of room temperature storage, in the cupboard or the kitchen counter, wherever it may be, convenient to remember to take it, is probably fine. That wouldn't be optimal for an absolute long-term storage. Like if we've had samples, for example, for research purposes that we've stored, not just in the refrigerator, but like in the freezer, and we've stored those for a decade, and then brought them out and did a study, and it works. And we've also done statistical modeling of shelf life in various storage conditions, and if you protect the astaxanthin not only from light but from moisture and oxygen, it can potentially, based on our modeling, be stable for decades. Like we predicted a median shelf life, like a 50% probability that it would still meet the label claim of the amount of astaxanthin in the sample, like 50 years later.
But in the freezer,
that was
in the freezer, right?
That was like a vacuum sealed bag.
But what about room temperature? Because when I buy astaxanthin, I don't know how long it's going to sit in the warehouses. What will be the concern in that situation?
Yeah. So generally, you have a couple 2-3 years of recommended shelf life, but that's because not only is it not protected from, it's mainly that it's not protected from air, and so. But if you were, say, to vacuum seal it or store it under nitrogen or argon, like inert atmosphere, so the oxygen is not getting to the astaxanthin, then it can last for much longer. But for normal purposes, if it's produced and it's sitting in a warehouse and then it's sitting on your shelf, if you're consuming it within a couple of years, it's gonna still be fine. And it's not like it spoils, like milk or something. If anything, the amount of astaxanthin might degrade over time. And so if the label claim was 12 milligrams, and we always include some overage when we produce it to start, just to make sure that we for sure have enough in the label claim to meet the label claim. And if it's stored for many years at room temp, exposed to the air, that it'll still be 12 milligrams through the shelf life best by date printed on the package. But even if you went beyond that, if anything, it's probably just slightly below the label claim of 12. Maybe it's 11.8 or 11.5 or 11 milligrams. And so that, from a shelf life standpoint, it's very good. I mean, if you wanted to have the optimal shelf life, yeah, you'd probably put them in a vacuum sealed bag and put it in the refrigerator, and you probably could store it for much longer. But once you bring it into your house and start taking it, it doesn't make a difference, because these things take time, and we've done accelerated stability studies looking at higher temperatures, higher humidity, and it can still be good, for the amount of degradation that you see over many months of time at those levels, supports that at normal conditions, it would be good for many years. So for the average consumer that's just going to buy it and use it, don't leave it outside in the sun, exposed open air, to the elements.
Do you keep yours in the fridge?
You just store it normally.
Yours you keep in the fridge?
I just keep mine. I just keep mine on my kitchen counter, so that way after dinner it's easy for me to grab. I normally take 4 capsules, 48 milligrams a day, and I'll just do that after dinner. For me, that's the easiest, because breakfast, lunch is not always as consistent in terms of timing, and so dinner is always a good time for me to take it. So that's what I do, and it works perfectly. And
you don't divide; you take everything at once. A bolus.
I do, but yeah, again, it'd be interesting to actually do the study of splitting morning and evening versus single dose. But I think either one is fine, and there's going to be variation between individuals anyway. So I think it's something that the most important thing is to take it and take it consistently. So whatever is conducive to doing that. If it's easier to split it or easier to take it all at once, I think that whatever allows you to take it consistently, that's going to be the best.
By the way, about the previous subject we spoke about, if you were to speculate, somebody's healthy, he took astaxanthin, how long would it stay in his body until it's going to be oxidized? What will be the actual life of the molecule in the human body that does not suffer from excess oxidation rate?
That's a good question. I'm not sure. I don't know on that one. But again, that would be really interesting to study, because your body is always producing reactive oxygen species, and your cells are facing those insults on a regular basis, just from normal energy production and everything that's happening, and you have endogenous systems to fight that. But those endogenous systems decline over time, and also you can ramp up the amount of reactive oxygen species or other free radicals based on whatever it may be, exposure, to bad food or pollution or stress, or
X-ray,
exercise and infections,
CT
and
scans.
X-ray, all these things, right? And so
Is there
all
good
these
evidence?
things
Yeah.
have an
Is there
impact.
good evidence that astaxanthin can protect us from X-ray, CT scan, and even flights? Is there a good evidence that it does?
I don't know of evidence in that area. I mean, the evidence is more related to UV damage from the sun, but I don't know that we've looked at those types of things. It would be interesting to see. I mean, presumably it might, but yeah, I'm not familiar with research specifically on that. Yeah. Mm-hmm.
Yeah, I mean,
yeah. So there's a lot of
UV
interesting. Mm-hmm. Yeah. Yeah. So I think it would be reasonable to hypothesize that, and it'd be cool to study that.
By the way, because the astaxanthin, either formulation, it's absorbed via the fat absorption system that we have with the micelles and all of that. Do you think some of that also goes through the lymph as well, or it goes directly to the blood?
So it could be packaged with chylomicrons and then go through the lymph, but it also could be like an HDL pathway. And so again, that's areas that for carotenoids in general and for astaxanthin is being further explored. But yeah, it's ultimately it gets packaged with the lipoproteins and distributed. But that is an area that would require further characterization or elucidation, as we expand the research.
Yeah. Okay. Yeah. So in essence, yeah, we kind of gonna get small amounts. Just my imagination, small amounts. Some of that gonna go through the blood. Some of that gonna go through the lymph and entering the blood at some point. Let's say that. Now, I know that at some point it's going to be accumulating fatty tissues, like brain, skin, the eyes. From the time you take it, how long does it take it to go to the brain, the eyes?
Yeah, let's see. Well, in humans, we don't know that because we can't do tissue samples in humans. Yeah. And in animals, we haven't done, when we did the tissue collection and analyzed, it would be, for example, after 8 weeks of dosing, or 12 weeks of dosing, and we wanted to just measure at that point when we're looking at the efficacy results, could we correlate it with tissue levels, and confirm it got to certain tissues like the brain or the heart. And on the pharmaceutical development pathway, you can do things like radio-labeled, astaxanthin, which we have actually produced in the past and had developed for our pharmaceutical development, where you can actually dose it and then, unfortunately, sacrifice animals at various time points and look at where the label astaxanthin occurs, and you could see at different time points how it goes throughout the system in terms of absorption, distribution, metabolism, and then excretion, ADME studies as they're called. And so that's something that I think would be really interesting to know, but I don't know exactly how long it takes to get into the tissues. But it is something that from a anecdotal standpoint, people report, some people report benefits quickly, within days or hours or days, they feel better, whether that's placebo effect or real effect, who knows? And whether that's a systemic, result of it's in the blood, it's being systemically distributed, and you're getting some initial quenching of oxidative stress or reduction of inflammation that's having an effect. Versus how fast it's in the tissues, and not just how fast it gets to the tissue, because it's being distributed throughout the plasma, so some is getting into the tissue, but in terms of how quickly it's accumulating in the tissue and how fast you have a biologically relevant amount of astaxanthin in a target organ. And so those are things that if you know how much of the astaxanthin you need to have in a particular tissue to have biological effect, and you can correlate that, then knowing how long it takes to get there, that would be a really interesting answer. But that's, again, we wouldn't have that result. But practically, most people, if they are reporting some benefit that they can feel, because not everyone feels a benefit, like a vitamin, it's something that is helping you, but you may not feel it. But for a lot of people, they will feel a benefit, and it could be days or weeks or months, and again, that may depend on their absorption or their internal state of oxidative stress or inflammation, kind of what they're dealing with. So in terms of practically, I think days to weeks is probably an amount of time where you would start to have enough getting into your system.
It's not one of those supplements you take one day, you don't feel great, then this is it. There is accumulation that takes place behind the scenes that you need to take into account.
Yeah, and
what
it's
is
working
some-
at a fundamental
go ahead.
level, getting into cellular membranes and fighting off free radicals. And,
Yeah.
and so this is something that needs to act over time to really have the optimal results.
But my understanding it's correct. It's being accumulated more in the brain, in the eyes, and in the skin in humans.
Well, and the heart and the liver are, like when we've done our animal studies, those are very high accumulating organs. Like the liver it has a very large amount of astaxanthin that gets accumulated, which is a good thing. And we've done studies showing that it helps, prevent liver damage or reduce liver enzymes and things.
And the
Liver,
muscles
heart,
like,
brain,
yeah.
eye, skin.
And the muscles more like they have a lot of water and protein. Would you say that astaxanthin is not accumulated to the same degree in those tissues you mentioned compared to the muscles? Would that be correct?
Probably, yeah. I don't know. I don't recall if we have data on that in muscles specifically. I know we have, efficacy in terms of, like I talked about, in sarcopenia with muscle growth and strength. But yeah, you would hypothesize that there'd be more in the other tissues. And we certainly have found high levels in the other tissues. And we could certainly follow up on that. I don't recall exactly. But the important thing is that it gets, distributed systemically, gets to the major areas. And you've seen benefits in heart health, in liver health, in skin health, in eye health, brain health. Regarding the eye, though, I'll mention that lutein and zeaxanthin are actually, I believe there's like a receptor that brings them into the macula of the eye, and they're kind of like astaxanthin for the eye. Structurally, they're very similar. They're actually not quite as optimal of a carotenoid as astaxanthin, because astaxanthin has both the hydroxyl and ketone groups on the ring moieties at the end of the molecule. That gives it more polarity, allows it to better span and anchor across the cellular membranes, and provides less disruption, or doesn't disrupt the membrane as compared to other carotenoids, especially non-oxygenated like beta-carotene carotenoids. But lutein and zeaxanthin get importantly to the macula. Astaxanthin doesn't preferentially accumulate there like those carotenoids. And those carotenoids, lutein and zeaxanthin, the levels of those in your macula are inversely correlated with your risk for age-related macular degeneration. So as the levels of those go down in your macula, your risk of age-related macular degeneration goes up. And there's been major studies with supplements containing lutein and zeaxanthin and some other antioxidants showing that it can have an important benefit on age-related macular degeneration. And so with astaxanthin, it's not necessarily accumulating, in the eye, or at least not in large levels, but it's probably more the systemic, effect that it's benefiting the eye. Because there are eye improvements, visual acuity, tear production, reduction of ROS in tears and things that have been demonstrated. So there certainly is an eye benefit, but I think there are those other carotenoids that also are great for the eye. And with astaxanthin, I think that most of the benefits would be certainly lifespan as an overall health longevity, but heart, liver, brain, skin, those are areas that for sure you'll have, a lot of data supporting its use.
You have very good brain power.
Oh, thank you. It must be the astaxanthin. But no, I mean, I've been living it for a long time. So yeah, it's always fun to chat, especially with someone that wants to actually dig into it a bit more. So the fact that you're already up to speed on a lot of it is cool. So it's great.
I'm the consumer. I want to know what's the dose I need to take, and should I divide it? All those things, because I know the devil is in details. I know with my clients, there's a big difference between the dose. I personally think that low dose is going to give me radiation protection, but I think longevity activation, we need more of a dose. That's what my gut feeling. I'm pretty positive about it. I just need to pinpoint the dose. People ask me. I also publish my supplement routine with all the updates of the research. And I'm not absolutely sure. I put like 26 to 20. I'm going to experiment with 24 to 36 milligrams per day. And after the conversation with you, I feel it's right now. It feels like about the right dose. Made me think maybe I need to divide. If I don't need to divide, it supports the idea to take it with food, with fat, and no, with food, with meals. Technically, it should not be a fatty meal, right? I mean, just any meal is fine.
Well, a meal that contains fats, so you wouldn't just have an orange or something, But yeah, a meal that has fats in it, like a normal meal, is fine. It doesn't have to be a highly fatty meal, but just a normal meal with fat content, would be fine. And like you said, 24 milligrams, 36 milligrams. I I think we find that 2 to 4 capsules a day, 24 to 48 milligrams, has been the sweet spot for a lot of people. But I certainly think 1 or 2 capsules a day as a starting point is perfectly reasonable. And as a base case for maintaining health and supporting longevity, like we said, with the blood levels, that probably is putting you in the range of having those long-term benefits. But like we talked about, more is often correlated with, higher, with better results. So I think it's up to the individual how much they want to take, but also, like I said, individual variation in absorption. So I don't think there is going to be an answer for this is the dose. This is the dose to take. There may be an answer in terms of this is the blood level you want that we don't yet know. But I think it's what we could know is, or say is, this is the dose to start with, which we say take 2 capsules a day, for example, as a starting point. But you can start at 1 if you want, or start at 4 or 3 or 4 if you'd like, and then adjust. But give yourself 1 to 3 months. Look at things that you can assess or biomarkers, and then adjust accordingly. But otherwise, if you don't feel anything or measure anything, you're generally in good health, but you want to do everything possible to maintain good health and promote your health span and lifespan, then I think you can maintain a dose of anywhere from 1 to 4 capsules a day probably would be a good safe bet. But I think more research will be conducted, and we'll get a better understanding potentially of optimal doses or ultimately blood levels that are best suited for particular health applications or for longevity itself.
Any reason that you may think people need to cycle it? Maybe take 1 day off, 2 days off? Any indication about that?
No, I don't think so. I mean, because if anything, you want to just maintain levels and continue to, distribute it to your tissues and replenish it. And so it's not something like rapamycin where you have side effects or tolerability issues where cycling it may help you mitigate or get around some of those things. Or in the case where you're chronically inhibiting or chronically activating something in the cell, in a way that maybe gives you some type of benefit, but is not how your body was intended to function. So in those types of instances with other agents, cycling on or off may be necessary. But with astaxanthin, the way that it localizes in your cellular membranes, the outer plasma membrane, but also the nuclear membrane, the mitochondrial membranes, and the membranes of the other cellular organelles, it's just helping to support your endogenous antioxidant systems, fight off the free radicals, and kind of restore homeostasis in the cell. And so I don't think that cycling on and off with that is going to give you a benefit. So I mean, from a mechanistic, hypothesis, I think just having it on board consistently is going to give you your best result. With that said, you would have to actually have studies where you test consistent dosing versus cycling on and off and see. And I don't know of any research looking at that specifically, or anecdotally, I don't know of people having benefits that have tried cycling on or off. If anything, like my aunt, for example, told me that she stopped taking, she forgot to take it for a little while, and she noticed that her hips or knees weren't feeling as great anymore. And then when she started taking it again, she felt good again. And so that type of cycling I've heard of, but certainly not from a performance standpoint. Yeah.
Not taking is not a good cycling.
Yeah, exactly. Yeah. So
in my community of longevity hackers, they ask me about the impact on testosterone. I'm quoting the question. I've heard negatively impact hormonal function in men. I've come across discussion in Reddit that it may inhibit 5-alpha reductase, the enzyme responsible for converting testosterone into DHT. Anything that you know about the impact on sexual health, both men and women?
Yeah, so astaxanthin, I think, has been shown to improve testosterone levels. And that's thought to be by protecting the testosterone producing Leydig cells in the testes from oxidative stress. So I think it helps to promote testosterone production. But like you said, there is a potential inhibition, or at least research showing on the conversion of testosterone to DHT. And so I think more research needs to be done there to explore that. But I think that is what the research has shown. With that said, in terms of anecdotal feedback from our community, we've had reports of people saying that they have increased libido and sexual health and performance. And so I think I know that there are some discussions online saying potentially that someone had decreased libido, but then I think others commented and replied that that was not their experience. And so again, it's probably something where you have to look at what else are they taking, what else are they doing, lifestyle, all of these things to really know, scientifically, is it having an effect? But I don't know of any clinical studies in humans demonstrating a observable, manifestation of decreased libido or erectile dysfunction or anything like that. Because in terms of erectile, function or dysfunction, blood flow is important, and astaxanthin does increase nitric oxide and blood flow. And so, if anything, that could promote, healthy erectile, function. And we've had reports from people in our community that they have had benefits in those areas. So I think more research needs to be done in that area, but I don't think you can definitively say that it's negative, for male hormones necessarily. Certainly, it helps to protect the testosterone production at the testes. And also, if you look at things like
the conversion to DHT, which I don't see as a huge problem because it also affects male pattern baldness as well.
Mm-hmm.
But have you seen evidence that it affects this conversion from testosterone to DHT?
I've seen some published literature in that area, but again, I don't, again, is it replicated? How well is the study designed? And so,
it's like one study or something?
There is research out
small
there.
Data.
Yeah, there's not a whole lot of data in this area. So, so it's out there, but again, I think more research would need to be done, in particular in humans, to know, what is really conclusive here. But in terms of, say, reproductive health, which is not exactly the same, but if you look at sperm quality and performance, you see benefits there with astaxanthin supplementation. So the sperm linear velocity, the oxidation of the sperm, lipid membranes, the mitochondrial function in the sperm, all these things have shown improvements with astaxanthin as well. And certainly in the wild, or in, like, say, farm raised salmon, it does promote the reproduction and development. It supports that, and so I think that from that standpoint, it's been shown to be beneficial. And then with women, it is shown to reduce oxidative stress, to promote hormone health in women as well. So, with all that said, that is an area of research that does require further, exploration to really understand further. So, but I don't think you could conclusively say that it's not good for male hormones, or specifically for testosterone and DHT. But that's, I think, the extent of what is known. There's still a lot to be understood, even though there's, like I said, 3,000 peer-reviewed papers and 100 clinical studies. But you really need to do a lot more research to have definitive conclusions about these things.
Yeah, so not enough data. We don't know exactly. Yeah, we don't have good evidence to say that at this point.
I would like to add a few things. It's important to note that David rightly separated between testosterone levels and DHT levels. DHT, in case you don't know, it's a more potent form of testosterone. And yes, astaxanthin repetitively, in my opinion, has been shown to reduce this conversion from testosterone to DHT. The question is it really bad for you? Let me tell you my thinking on this. So to me, looking younger than my real age means that I need to preserve my hair. And we know definitely that DHT levels is a major contributor to male pattern baldness. So for me, even if there are some benefits to having higher levels of DHT, the trade-off makes sense. And second, here, as you can see, testosterone is being converted. So this lack of conversion may actually lead to testosterone preservation because it's not being converted into DHT. I've tried to double check this in healthy humans, not in sick animals, and I haven't seen any studies besides one. In this study, they gave humans a combination of saw palmetto, berry lipid extract, and astaxanthin. What did they find? A decrease in DHT, but an increase in testosterone, suggesting that indeed this lack of conversion preserves testosterone. I'm quoting from them. ANOVA-RM, which is a statistical analysis of data in which the same subjects are measured multiple times under different conditions over time, this method, I'm quoting, showed significant within-group increases in serum total testosterone and significant decreases in serum DHT from baseline. So it could be that astaxanthin actually preserves testosterone by preventing it from becoming DHT, which can actually cause you a hair loss if you're a man. But as David suggested, it's too early to conclude. We need more data, and I'm going to update you as more data comes.
Okay, let's speak about risks with brands. The first product that popularized a bit astaxanthin, I think, it has been krill oil. And when you go and shop for krill oil, you see different products with different levels of astaxanthin. One with 0.5, one with 1 with 2 milligrams per capsule. What's your take on the use of krill oil as a astaxanthin supplement? And do you see any risks in oxidation in the process of packaging the krill? What do you think?
Well, I think krill oil contains astaxanthin, but like you said, it's typically sub-milligram amounts, microgram amounts of astaxanthin, unless they specifically add more astaxanthin to get it up into the milligram amounts. So I think first off, you're not going to get enough astaxanthin from the krill oil. Not to say that krill oil is bad, but it wouldn't be instead of taking an astaxanthin supplement. So that's the first thing. And then in terms of oxidation, I would think having the astaxanthin in there would help prevent the oxidation of the krill oil to an extent, but at the same time, you don't want the astaxanthin being an antioxidant in the krill oil. You want to be an antioxidant in your body, and that's actually why in our formulation we have small amounts, not biologically relevant amounts, but amounts for shelf life purposes in our formulation, along with the starches and things like we talked about, to act as sacrificial antioxidants on the shelf. So if there is oxygen exposed to the product on the shelf, which it is, the vitamin C and vitamin E are there to sacrifice themselves so that the astaxanthin can remain intact to be an antioxidant in your body rather than on the shelf. So going back to the krill oil, again, it depends on what else. Do you have other antioxidants that can help to prevent oxidation, or also what are the production methods used to extract and store the krill oil? Because I know especially with the fish oils, there can be a lot of variation in the oxidation. And if you look at supplements versus prescription forms, that can lead to a really important difference in terms of whether it's actually good for you or bad for you. So I think the handling, the processing has a big thing. And then also if there are other antioxidants like astaxanthin in there, maybe that prevents the oxidation, but then now you're no longer getting that benefit in your body. So I'm not an expert in that space. But the main thing I would say is, if you want to take it, I would research, hopefully a good quality supplement for krill or fish oil or omega-3 source, but then certainly take, a good astaxanthin supplement as well.
Also, fish oil as well could be oxidized in our bodies once we consume it, because we do consume high levels of oxygen. In my mind, I would expect astaxanthin to be protective of our fish oil supplement. What do you think? Especially when it's just the DHA sits on the membrane. Do you think the astaxanthin are going to protect it?
Yeah, I would think so. And so certainly you think about the omega-3s helping to replenish the lipids in the membrane, and astaxanthin helps to protect those lipids from oxidation.
It gives them
and if you were to,
stability. That's the problem that they have. Yeah, they're very good for us, but they're also unstable.
Yeah. So if you were just trying to deliver the least oxidized form of fish oil, having astaxanthin in that fish oil supplement would presumably help to mitigate the oxidation in addition to proper handling.
But if you take it separately, even
but
if you take your product, go ahead.
Yeah. So you're talking about shelf life versus what's happening biologically. Yeah. So I think there's considerations for both. Again, I don't know that there's research in those areas, but I mean, you can hypothesize about that for sure.
Yeah. So if I'm 65, I have above average oxidative stress. I'm taking fish oil. It sits on the membrane. After 2 days, it begins to oxidize. I would expect astaxanthin to come and stabilize the membrane. Do you think it's a good scenario?
I think that's reasonable. Yeah.
Because that's really important, especially to me. The greatest impact of astaxanthin could be with the elderly, because they lose hearing, they lose eyesight. It tells me they lose brain tissue. It tells me they oxidize their fatty tissues. This is why the way I see it.
And so I think it definitely can help the elderly, but then the question is, you don't want to necessarily be starting it when you're elderly. I think that it has benefit. Certainly, like we looked at geriatric animals and shown benefits there, and so I don't think it's something where it wouldn't work in the elderly. But the whole idea is just, if you can start taking it younger or as a normal age adult, and hopefully maintain better health, then by the time you are elderly, you're in a better state of health versus trying to fix things that have gone awry. But to your point, I mean, yeah, those are the things that over time, the oxidative stress, inflammation, that's happening over years and decades takes its toll. And so anything you can do along the way to mitigate that is going to be a big help. And then once you are at that point, anything you can do to help the decline or help to maintain or restore, is going to be beneficial.
Yeah. I have a few questions about the products from my Longevity Hackers group. They ask, what's the risk of consumer buying astaxanthin from brands? Any risk that you would warn consumers?
I think you would want to just purchase from a reputable brand that's that is known in the space to be credible, and so. Just to rattle off a few brands, like for example, BioAstin is a form of microalgal astaxanthin grown in Kona, the Big Island of Hawaii. Their company was literally across the street from our company. And so our company and their company were the first two astaxanthin dietary supplements on the market back in 1999-2000 timeframe. And so they've been around a long time. They've done a lot to contribute to the growth of the astaxanthin market. And they not only sell their BioAstin brand, but they supply their ingredient to a lot of other manufacturers or other brands that sell it, in as part of their catalog. So their product is certainly one that has a lot of history behind it. It is grown in the open ponds, and so it is potentially more prone to contamination versus a closed pond approach. But with that said, I think their product is, of good quality and is reasonable, to take in terms of if you want a known, trusted source of microalgal astaxanthin. But there's also other forms of microalgal astaxanthin from companies like Asta Real or Asta Real. I've heard it pronounced both ways, but they're based, their U.S. Headquarters or production facilities are in Washington State, and they have an entirely closed tube production, and they use hydroelectric power that's sustainable to power the production, and so that's the closed system, so potentially less prone to contamination. Also in Iceland, Algalife produces microalgal astaxanthin in the closed tubes, and they use geothermal power, which is also great. And so theirs would be a nice, clean product that's environmentally sustainable. But with all of those sources, again, you're still getting the microalgal form of astaxanthin, like we talked about, that would not be as bioavailable as our form. So there is that consideration. But I think if you are looking for the microalgal form, those are all trusted manufacturers. And then there's brands that are not manufacturers, but they are credible brands for supplements and vitamins in general. So if you look at Amazon, Sports Research is a brand that sells astaxanthin. In the past, they sourced their astaxanthin from Asta Real, and then they, I believe, switched over to Algalife, the Icelandic form, but basically the same astaxanthin closed, tubes. So whether you do the open air ponds, like the farmed, BioAstin markets it as kind of like a farmed astaxanthin, Hawaiian farmed astaxanthin, or the closed tube systems like Algalife or Asta Real. I think those manufacturers and the brands that they sell to are more than reasonable in terms of credible and to be of high quality. I know that there was that study that showed that some brands on Amazon had significantly less astaxanthin per capsule than the label claimed. So that's a consideration. But I think if you are purchasing from one of those,
so there is evidence.
It does happen. Yeah, there is evidence for that. That was not us. That was some other. I forget who the group was, but they published a report on the top number of products on Amazon, and then they analyzed the astaxanthin content. And again, astaxanthin is tricky to analyze and measure, and we spent a lot of time and money developing methods to properly analyze it. So I mean, you have to make sure that the analytical methods were robust.
So I see here. So to me, the risk here is that the retailer may buy from the manufacturer, and since he doesn't have the technology to measure properly, he cannot keep the feet to the fire of the manufacturer. Is going to take their word because it's so complicated.
It's possible. Yeah, I mean, there are published methods, though, for just measuring the amount of astaxanthin in a product sample. It's a little bit more tricky when you get to measuring it bioanalytically in the plasma and tissue. That's especially where it gets more tricky. From a dosage standpoint, that is a little more straightforward. And so I think as long as the manufacturer or the brand is a reputable company that is above board and trying to sell the right amount of astaxanthin as labeled, then you could be reasonably assured that it's going to be the appropriate amount. That's certainly one thing that we do is are very rigorous about making sure that we have the labeled amount there. But yeah, so I think if you look at the top selling products on Amazon, those are probably more than fine. But if it's some upstart brand that just is on Amazon and appears to be too good to be true in terms of how cheap it is or something, then I would probably be more skeptical of that. But so if I was doing microalgal, it would probably be either a BioAstin or Astareal or AlgaLife sourced astaxanthin, whether it's directly from them or from a reseller like a Sports Research. I think any of those would probably be fine. And then in our case with AX3, we call it AX3 BioPure astaxanthin. We call it BioPure because it's highly bioavailable pure astaxanthin. And by the way, we call it AX3 because it has 3 times the absorption. And so that's also why we refer to it and AX for
yes.
Yeah. And so, but yeah, so I think,
and this is a formula they use in the ITP, right? This is a formula they use in the ITP. So, and it includes three types of astaxanthin, isomers of astaxanthin.
Correct. So it's the same. Yes, the same. So it's the synthetic or astaxanthin produced by natural product total synthesis. So it's synthetic. It's non-esterified. It's the mixture of the isomers, and it's the exact same formulation composition with the starches and the glucose syrup and the exact same formulation. It's of course not in the capsules, but it's the formulation, the beadlet particles, which is the starchy, dispersed, finely dispersed astaxanthin in the starchy matrix. That formulation was incorporated into the chow that was fed to the mice in the ITP.
I think one of the risks, in essence, is the variability. So the person may not know if they buy the algae-produced astaxanthin, how much, what's the blood level they're going to get. That will be like one of the greatest risks. Or buying from a very cheap manufacturer that has less astaxanthin than they claim. So that will be like the main risks. I would say. Is that fine to say that?
Yeah, I agree. And then the one other consideration is just not a risk, but a consideration from a price point. If you look on Amazon at, say, those top-selling microalgal astaxanthin brands, you'll typically see 60 capsules sold for around $25 to $30. And then if you were to look, say, at our product, you'll see that it's 60 capsules of the 12 milligram capsules for $50. And you'll think, to the untrained, consumer, it'll be like thinking, wow, we're double the price. But if you think about needing 3 times as many capsules to absorb the same amount, given our head-to-head human study, then presumably you would need to take, 3 bottles to equal one of ours. And so that would be something where you'd be looking at spending $75 for 3 $25 bottles to equate to one of ours if you extrapolate the results of that head-to-head PK study, where from the same dosage of 12 milligram capsules in the microalgal versus our form, you had a 3 times difference in bioavailability. So that's one thing that when shopping around, I think people should also consider. But just to reiterate what I said before, I think at the end of the day, if people at least just supplement, their diet with astaxanthin, that is a win. That is a good thing. And in addition to all the other lifestyle, diet, exercise, sleep, stress, all these things that you would do for a holistic approach to, improving your health and longevity. So this is not to replace any of those, but I think in addition to that, and then I think just taking astaxanthin is already a great thing. And then we have our reasons why we think our form is optimal. But we certainly have experience working with the microalgal, and potentially will offer microalgal as part of our product line in the future as well, just to give those options to the consumers. But those are the considerations that I think a health conscious or sophisticated consumer should think about.
Let me just share my enthusiasm about astaxanthin. My enthusiasm about the supplement really grew when in June 2024, a Yale's aging test was published, and I've done the aging test, and I noticed my brain wasn't on the same rejuvenation protocol like the rest of my body. So I realized I need to target the brain specifically, and astaxanthin is one of the only supplements that can do that. The other option is hyperbaric oxygen. In addition, I experienced a lot of chronic psychological stress because of my wife's situation, and I also suspect it's going to contribute to brain aging. And I knew also because of my wife research that I had to do, that very difficult to get into the brain. And astaxanthin is one of the only supplements that actually can target the brain. And if my understanding is correct and it actually activates longevity genes in the brain, maybe it would fit into my overall strategy. But I want to test that with, I'm going to keep the audience updated, but I want to test that because I have seen with the hyperbaric oxygen, it reversed a few years back in my brain. And we know it targets the brain. So I would be very curious to see how it's going to affect my brain as well. And also I'm curious to see maybe it could help my wife with her stroke as well. That would be a good experiment as well. I wanted to tell the audience about your website and maybe if you have different versions of the products. If you have a variety of products, maybe forms, maybe different bottles, please feel free to share anything that you want about your product. And also the website, of course. I'm going to put also in the description the links to his products as well.
Yeah. So, well, first off, I think the study that you're doing sounds really interesting, and it would be cool to see what happens with the brain aging. Of course, I think that there are different biomarkers for biological aging and organ aging, and so I think there are various considerations on how that maps to the reality of the aging in lifespan over time. But I think that that's the best tools we have at this point, right? In humans, and it's really cool to see, especially on on yourself, what results you're getting, and then with your wife. Again, so sorry to hear about that, and I do hope that Astaxanthin could help her, with her health going forward. So with our website, it's ax3.life. So not .com, but .life, and hopefully for increasing your lifespan and your health span. And so on Instagram, it's also ax3.life. And right now, we just have our single form of the product, which is the 12 milligram capsules. And this is with the BioPure, the synthetic, highly bioavailable formulated version of Astaxanthin. And we sell it in environmentally sustainable pouches that use less material than a plastic bottle and are much lighter than, say, like a glass jar or bottle that would take more fuel to transport. So we're trying to be sustainable in terms of the packaging. And so that's what we currently offer. In the future, there are plans to release a powder form that you could disperse into water. Like I said earlier, you can already take the capsule and open it up and put the powder into water or a drink, like a shake, and consume it that way. But we are looking into releasing
as long as it's with a meal.
Yeah, with a meal. Yeah, so not instead of a meal, but along with a meal or after a meal. Exactly. And so it will turn the water bright red, which is strange. But it won't taste or smell differently. So it's an interesting experience. But that's an option. But we are looking at a powder form where you could just dispense the powder, directly in without needing to open up a capsule. Potentially
Would it reduce the cost?
Form.
Would it reduce the cost because you're going to save the capsule? Would it reduce the price? If somebody out there, it doesn't want to just take 12 milligrams. I said, I'm going to take 24, 36. You're already taking 48. A person may decide based on their individual choice. It will try to find a way to save money, maybe to buy in bulk. What would be, so that's like, that would gonna save money if they're gonna buy the powder in bulk, in a more bulky way?
Yeah, potentially. And so we have to still figure out the best packaging. So because it's a pigment, so it's something where if you are scooping it and it gets onto the counter or onto your clothes, it potentially can stain. And so we have to have considerations about, whether it's a bulk amount of powder that you're scooping. And again, the amount that you're scooping is going to be very small. It's not like you're scooping like a protein powder or nutritional shake powder where you're having big scoops. This would be very small scoops, even if you're doing the equivalent of multiple capsules. So that we'd have to figure out if you're scooping powder or if it's individually packed amounts that are in like environmentally sustainable little packs that you can tear open and put into a drink, kind of like you might with an electrolyte type of mix, for example. So those are the considerations, but to your point, it's not needing to be encapsulated, but just packaged. And so the cost of goods could be less, and therefore we could pass on some savings to the consumer and potentially offer a better price point, especially for that power user that wants to purchase in bulk. So that's a future plan, but for now, you can get the capsule and dispense that into a drink or just obviously swallow the capsule. We are looking at gummy forms for the future, which from a health standpoint, to make a gummy, you're incorporating other ingredients, and that is typically not optimal, especially if you want the gummy to taste good and have shelf life. But again, it's probably better to take astaxanthin even in gummy form than not at all. And so if the gummy form makes it fun and good to take and you enjoy it because you have a nice taste, I think that's still a good benefit. And so that is a future plan potentially as well. And we may look at a pet formulation because, again, right now you can take your capsule and put it in peanut butter or cheese or soft food and give it to your dog, for example, or people sprinkle it into cat food. Other pets, I've seen that people have given astaxanthin or AX3 to them as well. But having an actual little pet treat or chew could be a nice way to make it convenient to give to your pet, because obviously we'd all love our pets to live longer, healthier lives. So those are all plans. In addition, we are looking at, even though we have scientific advantages and manufacturing advantages over the microalgal form, we are looking at adding a microalgal form as well, just so that people have the option if they would prefer that, even if it's not as well absorbed. So those are all future plans. At this point, we just have the encapsulated form of the BioPure synthetic, highly bioavailable formulation of astaxanthin. And so again, that's AX3.
Can somebody subscribe and get some discount if they subscribe to a monthly? Do you offer some subscription at the moment?
We do. Right now, we just have it actually as subscription only, because this is not something you just take once and then you're cured. And so it's just offered as a subscription only. And so if people don't want to subscribe, they could always cancel.
You ship worldwide, or you ship only America?
We ship to Canada. Well, the United States, Canada, Australia, New Zealand, the UK. And a few other areas, not currently to the EU, but we are looking to expand in the future there as well. So not completely worldwide, but certainly more than just the United States.
I'm curious, like if I take 3 capsules of your formulation, how many grams of carbs would I get? Like half a gram of carb or something like that?
Yeah, so the modified food starch, that is the main substrate of the formulation, is about 180 milligrams per capsule, so less than 0.2 grams. And so if you had 3 of those, then a little over half a gram of that. Okay. And then the cornstarch is, not significant. The cornstarch is about 50 milligrams, so 0.05 grams. And so altogether, if you add that together with the modified food starch, and by the way, the modified food starch is not genetically modified. Some people think that means genetically modified. It's modified where it's processed in a way to make it where it's has better water dispersibility characteristics compared to the normal starch. And so that is what actually allows it to be so well dispersed in the water and therefore in the GI fluid and then nicely absorbed. So that's what the modified starch is. And then, but if you take that plus the very small amount of glucose syrup, less than 1/10 of half a gram per capsule, it's a very small amount of the starch or carb and sugar content in there. So it's I would say negligible from a dietary standpoint. But it is
about half a gram or more, just
formulation.
To get a number. I like to know numbers. Like
approximately half a gram.
Yeah.
If you take 3, it'll be a little more than half a gram, but it'll be well less than 1 gram.
Okay. Anything else you want to share? Maybe new studies that you're working on. Maybe preclinical data that hasn't been published yet that you want to share?
Mm-hmm. Well, the ITP is based on the results of our published study, are really excited, and they're testing it again, but now they want to see, well, what if we do a significantly lower dose? Can we still have a lifespan benefit?
Oh, really?
And so that'll be interesting to see. Yeah, so they've already started that. We've supplied them with more material. And so, let's tune back in 3 or 4 years, right, and see the results. But so we'll see. If anything, we were also wanting to maybe see what if we did a higher dose. Could we have more
what I'm interested
lifespan benefit?
because my understanding of what you said that is what they did equivalent to 12 to 24 milligrams in humans. So so I would actually, so practical perspective, I mean, people can take more than 12 milligrams. I would want to see double the dose. That would be
so
much
what it
more
comes
interesting.
down to, I agree. Yeah, so we pushed to do that. They wanted to test the lower dose just to see, kind of to get that scientific answer. But from a practical standpoint, the reason why the higher dose wasn't feasible is you can only incorporate so much of another ingredient into the chow and still have the chow be intact. And so the percentage, that the concentration of the active ingredient in the chow is something that is limited, and so we're bumping up against that when we start to have the higher dosing. So that's
they already had a problem.
It's just hard to get it in.
They already got
they were
like
already
60% less.
exactly. Variability there. Yeah, so that's the thing.
You probably need to serve them some drink next to the meal, force them to drink it with the meal, and then take the glass.
There you go. Yeah, so I mean,
take them to a bar, a Astaxanthin bar, drink.
And then they also may in the future look at, their primary study is the lifespan study, where they just start the mice with dosing at some point. In this case, they were 12 months of age, which is the human equivalent of being in your 40s, based on these mice. And so they started middle age. Other interesting answers would be starting later age to see how much of a lifespan benefit you can, or younger. Either way, it'd be great to have those questions answered. But also they have another variation of the study where they don't just look at the lifespan, they actually look at health span, like tissue, effects, organ effects. And so they'll take animals at different time points and all those things. And so that would be another follow-on study that would be interesting to see. But at this point, what they're currently doing is just the follow-on lifespan study at the lower dose. So I'm not sure how that's going to turn out because the dose is significantly less, but it'll still be an interesting contribution to the science.
I think last question: are you aware of females dying from different cause of death compared to the 80% mortality rate by the male mice? Do you know any differences in the cause of death between these two groups the ITP uses?
No, and I don't know that they have the cause of death specifically characterized in those. I think that's where you look more at the health span studies. That's where they really get into what's happening, and both in the progression of issues at the organ level and then ultimately cause of death. And so all they're looking at in the main ITP study is just how long they live. So we don't have the exact answer. So so again, wouldn't know exactly if there was a difference for cause of death. I would presume not. I presume it's probably similar causes of death. But that again would be interesting to know with further research.
Okay, I think we can go speak Astaxanthin in another hour and a half or so. But we've been going for a while here, but,
I
mean, it's quite amazing how much you can speak about it.
Oh yeah.
Okay, let's repeat again about your website.
Sure. Yeah. So it's ax3.life. That's the website. That's it. And we just have the single product there for a subscription, and people can do 1 to 4 pouches per either 30, 60, 90, or 120 days, whatever is the most convenient. And the subscription can be adjusted at any time. There's an account that you can log into. You can skip, reschedule, cancel anything, change the quantity, however. So there's complete flexibility. And if someone really wants to just try it once, which I wouldn't recommend because I think you want to take it consistently. But you could always purchase and cancel, or you could purchase and extend the frequency of orders to 12 months and then manually trigger an order whenever you want it. So I mean, there's workarounds, but essentially we encourage a subscription with regular dosing for optimal benefit.
Perfect. I'm going to put a link as well in the description. So I think this is it. Thank you so much. And thank you for listening as well. I don't think I've ever heard such a long conversation about the Astaxanthin. I think it deserved the attention we gave it, and I think we could go. I mean, it's such an interesting molecule. We could go and speak even 1-2 hours. Hopefully, more longevity studies gonna come out, and we can maybe gonna give more specific directions about doses, cycling, not cycling, etc. So subscribe to the channel. So thank you so much, Dave.
Thank you. Yeah, we're just getting started. Thanks for having me.